achieving is to identify all researchers, academics, students working on genomics and bioinformatics. First, I would say within Nnamdi Azikiwe University, Awka, and other participating institutions around the Southeast geographical region, irrespective of discipline. And from the abstract we have already received for this ongoing symposium, it's already clear that there are people, even in the sister university, even in Delta State, even in Enugu, who are working on bioinformatics. So I believe that the conference and this symposium will be able to identify these persons so that we can be able to enhance and collaborate together and probably embark on a very serious genome-wide project like Igbo Genome project, which currently there is no data on particular tribe known as Igbo. So, and I believe that this consortium at the end of the day will be able to have these projects going on. Now, also at the time we were starting, it's also in our mind to organize conferences on bioinformatics and genomics, of which you are in now, so that we can attract other participating institutions around Awka and Southeast region and Nigeria, also to help identify other investigators who may not be affiliated with the local Nnamdi Azikiwe University. So we also aim to collaborate with other bioinformatics interest groups in Nigeria, Africa, and internationally. So in Nigeria, of course, the ProSeq Bioinformatics, Noun Data Led Africa, and also the H3 Bionet, and all of that. So these are the guys that we are already collaborating with to ensure that we advance the course of bioinformatics in Southeast Nigeria. Yeah, so for the funding sources of the consortium, of course, we have members ranging up to 200 who in the course of our training, they have highly subsidized training and also make commitment of 2,000 annual dues yearly. Then we also get grants for training from the university and from other bodies, and also for projects. Yeah, we have partners in this game, and the one is the wet lab partner, so the Molecular Research Foundation for Students and the Scientists, which is more of wet lab. And also we have the ProSeq, I said, which I think the correct name is now already matched with Data-Led Africa. The H3 Bionet has given us the opportunity to host what we call IBT training, Introductory Bioinformatics training, for 2 consecutive times now in 2020 and also in 2021. The consortium has been able to host these classes, building capacity locally so that people will begin to gain interest in the area of bioinformatics. So capacity building, we actually started with this first to engage the environment on what essentially is bioinformatics. So we have to start with what we call the entry-level bioinformatics workshops, which we had July 31st through August 2nd, 2019 in Nnamdi Azikiwe University, Awka. So we called it entry level because we needed to get a lot of people who don't know what essentially is bioinformatics and start with what is the definition of bioinformatics, and also move on from there onto databases and other things that are involved in bioinformatics. So we went on also to, in NGS, this was locally organized, and many people also got involved. So as I am making this presentation right now in NIU, there are a lot of persons that have these skills to be able to analyze NGS data coming from PacBio or Illumina or some other platform, as the case may be. So during the COVID, of course, we are shut down, but we are also still ongoing. So we are able to run some program on galaxy bioinformatics. Yeah, so this took some days, but I think at the end of the day, that was also wonderful. Also, we had the molecular docking, which was an online and on-site program that we also had together in the course of the lockdown, and the outcome was also awesome. This also need to be connected to the molecular dynamics, but we decided to break this and fragment this into the docking, then the same group of persons and those who already have some skills in the molecular docking to also move on to the molecular dynamics. Unfortunately, maybe because of this particular conference and symposium, the dynamics supposed to hold the same period, and also the resource persons are very busy for this symposium, and we decided to defer this probability next year. And we hope that the capacity building will still continue next year. So in the course of the year, particularly in 2021, we had a series of webinars starting from applications and the essential. So why do we need to do all of these in the NAU NJBC? The reason is that we need capacity in the Southeast Nigeria in bioinformatics. You know, without capacity, we might not even have resource persons that will be able to drive the course of this research around that environment. So in order to publish, in order to do what we call an outreach programs, so we started our webinar, which first was applications and the essentials of it. Then thereafter, we had the prospects, which one of our alumni guy also took, Dr. Tangod Ebenezer, in March 2021. So we had prospects in it. So all of this is to advertise and to sensitize people of what essentially bioinformatics is, and maybe why they could consider bioinformatics as a new career that they could advance therein. Okay. Then for those who are particularly interested in drug discovery and design, we also designed, a webinar to also inform that bioinformatics, of course, cut across many disciplines, including those in pharmacy and those who discover drug, who can learn on how to design ligands and also protein interaction that will help in determining the binding affinity and many other parameters that could give an idea that the drug or the ligand, could be a potential anti-infective agents. Okay. Yeah, so of course, we have been on course. We just ended the 2021 H3 Bionet IBT course in October. That is for 2021, but this started from 2022. And of course, our classroom, of course, held virtually, and many persons are on ground currently having these skills on bioinformatics. Now, also the molecular dynamics, which I've already shared about, that already been deferred into next year. And currently we are on our bioinformatics symposium happening right now and before us. Okay, just to say that as the coordinator, I'm not alone in NJBC. We have many persons, starting from the Vice Chancellor himself, who brought the idea with Professor O.C. Nwogo and also, thank God, Ebenezer, to initiate this particular NJBC. Then we're also happy to Dennis Arebodo and all the guys you are seeing their names here, who themselves are also working at the background to ensure that this current symposium is ongoing. Yeah, so the LOC chairman, who is Duru Vincent, and all of that. So I want to thank you, and also from my own table as the coordinator, welcome you in this symposium, asking you to protect the time for this workshop, for this symposium, protect the days, the 3 hours per days, and I hope we shall be able to get through, and you would have been inspired at the end of the day on the particular course to take and to push personally for this awesome, evolving career. So thank you. So maybe at this point, I will need to hand over to the technical chair of the day, who himself is Dr. Samuel, to to probably take over and introduce the keynotes for today, Nicola Muda, professor, and H3 Abayanet. So Samuel, I think over to you. You should be a co-host now. And yeah, thank you, Samuel. So Professor Nicola is also here already. So we are going to make her a co-host so that she could also share her slides after you would have read her profile and introduced her appropriately. Thank you. Yeah.
Thank you so much, Dr. Moses. I appreciate it. Can you hear me?
Yes.
God bless you. Thank you so much. I want to appreciate everyone that has come for this symposium. And I can assure you that for the first time, I believe this is a set of people that are sincere and they are very passionate about developing capacity in Nigeria. I've had the opportunity of working with this group, especially the coordinator, for the past 2 or 3 or 4 years now. And I've also been on site in Nnamdi Azikiwe University, and I know that a lot is going on there. Okay, so without much ado, I also want to introduce someone that I've known also for a couple of years now. I've known Nicola since 2008 when I first came to South Africa. I was with the ICGB and I moving around the university campus, the IIDM, they call it then. And I stumbled into a department and I fell in love with bioinformatics since then. And she has also, because I registered for a training and she was one of the people that facilitated that training. And that stuck to me. She's a very soft-spoken person and very easy to understand. She taught of biological databases and I've been using databases since then. And so Professor Moda heads the computational biology division at University of Cape Town. And is a full member of the Institute of Infectious Disease and Molecular Medicine. And she leads the H3A Bionet, which is a large pan-African bioinformatics network for 28 institutions in 17 countries. I was also opportune to be part of that, which aims to develop bioinformatics capacity to enable genomic data analysis on the continent. So the H3A Bionet has developed an extensive training program for African researchers, and I mean, you just alluded to one of that being done in UNIZIC. She also leads a sickle cell disease data coordinating center and a Wellcome Trust Center data integration platform at UCT. She received a PhD in microbiology from the University of Cape Town, then worked for about 8.5 years at the European Bioinformatics Institute in Cambridge as a team leader. At UCT, our research focuses on genetic determinants of susceptibility to disease, African genome variation. I shall publish a lot in that, microbial genomics and infectious diseases from both the host and pathogen perspectives. Our group provides bioinformatics services and training and develops new algorithms and resources, and of course, a lot of tools for the analysis of complex African genetic data. Professor Moda is actively involved in capacity development, including training, education, and curriculum development in bioinformatics. She also sits on a number of institutional scientific advisory board. And I can, with pleasure, want to welcome Nicola Moda. Recently, I got a good news from the person you recommended to me, Emil, that the grant was granted. I'm so excited. I'm so excited, Nicola. And it's an opportunity. It's a great pleasure to have you here. And I believe we are all ready to listen. Please, I request that you please mute your mics. Let's give the utmost attention to this presentation. And host also, I noticed that when there is a sound, if it's possible to mute that sound when people log on, if it's possible. If it's not, then it's fine. Over to you, Nicole.
Great, thank you, Moses, for that introduction, and thank you for the invitation to speak today. It's nice to see you again, even if only virtually. So I'm going to be talking about bioinformatics capacity development in Africa. And so I'm going to talk a bit about the research that we're doing in my group at CBio. just to give you an idea about the kind of the scope of potential bioinformatics applications in health. And then I'm going to talk a bit about the big data challenges, and then how these consortia, H3 Africa and H3 Bionet, have been trying to deal with all the data needs and addressing some of these challenges, and then focus particularly on the human capacity development. So in my lab, we use bioinformatics for various different biomedical research questions. So we focus on two things, both the pathogen and the host. On the pathogen side, obviously there's pathogen genome sequencing and annotation that can be done to really understand the biology of the pathogen itself. More recently, obviously, with SARS, there's a big focus on pathogen surveillance and epidemiology. So tracking the NGS to track the new variants and to look at transmission dynamics. One of the things we've done in my group also is to look at functional interaction networks. So that is within a pathogen. So for example, within TB, to understand how all the genes and proteins interact with each other, to better understand the biology of the bug, how it causes disease, and which proteins or genes would be good to target if you wanted to try to find new targets for new drugs. And then we also have done a project. We're looking at analysis of different strains of TB. Excuse me. These are clinical isolates from South Africa to really understand the path of virulence from the genetic, genomic, transcriptomic, and proteomic perspectives. We then look at both the host and pathogen together. So we've had projects that have been modeling transmission of TB from the host. So this is looking at people who could go to different settings in poorer areas. They have GPS devices, and we model how the TB gets spread from one person to another. And then, because we have data from both the pathogen and the host, we've been looking at host-pathogen interactions. So this is looking at where particular proteins from the bug interact directly with host proteins, but also where there's indirect interactions, like the bacterium has some sort of effect on a host pathway via an indirect rather than a physical binding. And then we also look at the host itself. So we know that the African populations are extremely diverse. Excuse me, I have this annoying cough. The African populations are extremely diverse, and therefore actually very interesting to study, but really necessary to study, because they're the most ancient populations, and so we can learn a lot from studying at a very granular level the diversity across different African populations and the relevance connection to disease susceptibility and response to treatment. So we do a lot of population genetics. We look at genome-wide association studies. We do NGS and GWAS analysis, post-GWAS analysis, and we build tools for working with African human data. And then finally, we look at the microbiome. So this is different components within the host that impact disease. So we look at the gut microbiome, the genital tract, the nasopharyngeal microbiome, and many other areas where there are potentially sites of diseases. So, from what you can see here is that bioinformatics can be used, if you have the right data, bioinformatics can be used to tackle the entire system of both the pathogen and the host and how they interact to cause disease in order to better understand infectious diseases. So I hope this has given you an idea of how one can use big data in life sciences. It enables novel discoveries for healthcare, but also for agriculture, food security, disease surveillance, etc. And we've seen that with COVID-19. I mean, the data sharing in the COVID-19 pandemic has been unprecedented and really has enabled kind of the world to try and stay on top of this pandemic. And then there's also management of health data. So, we work particularly with the genomics or NGS data, but the clinical data coming directly from health records or from research cohorts can also inform diagnosis and treatment to enable precision or genomic medicine. And so what it means is that when you analyze big data that's relevant to health, you can actually get real health benefits. So there's a direct consequence of the correct interpretation of the data. But there are many challenges. So Previously, I mean, this is many years ago, we had just a little bit of data, but that data was very well understood, very well curated. So we were quite rich in information. Now we have so much data and very little of that has really been explored to its full potential. So we have too much data and not enough information, which should be converted into knowledge. So the big data needs, so the big data is really defined by the 3 V's: volume, velocity, and variety. The generic challenges in big data, and I mean, variety is really the one that's the most relevant, I think, in bioinformatics. When you think about astronomy data, our data is not that big. The speed at which it comes is, again, not that big compared to, say, the data streaming in from telescopes. But it's the variety and heterogeneity of our data that makes it very complex. So we have challenges of data capture, data transfer, data storage, analysis, interpretation, finding the data, visualizing the data, and then eventually sharing the data so that other people can use the data and that can actually inform policy. In Africa, of course, we have unique challenges. These are the internet, power. We have in South Africa, we have these scheduled power cuts. And so when you're trying to analyze data or when you're running big, long processes, these just get interrupted. Generally, there's limited computing infrastructure, limited skills, and in some cases, limited data. So only now we're starting to generate a lot more genomic data for African populations. So one of the consortia that was designed and developed to address some of these challenges is the Traffic Consortium. This stands for Human Heredity and Health in Africa. And this is funded by the African Academy of Sciences now through the Wellcome Trust and the National Institutes of Health to basically build a consortium of research projects that look at the genetic and environmental basis for diseases in African populations, but also that build the basic research infrastructure, build collaborative networks, and do capacity development. So the consortium includes about 34 different countries and many different H-Africa projects. And these are studying a variety of diseases, from cardiovascular related diseases to monogenic diseases, mental health, and then infectious diseases. Now, across this consortium of diseases and projects, they're generating data and biospecimens for up to 100,000 participants across Africa. Now, that's, again, unprecedented in terms of the scale of data collection on genomics in Africa. So this brings major data and bioinformatics needs. So we need computing infrastructure, data management support, analysis, storage, submission, access. And so we needed a full infrastructure for, to build this data ecosystem, and then training in each of these components. So H3 is the Pan-African Bioinformatics Informatics Network that was established as part of H3 Africa. As Moses said, it covers nearly 30 partners in 17 countries, 16 of which are in Africa. And H3 Bioinformatics has been working on developing bioinformatics and data analysis skills, the infrastructure itself, and then various different tools. So you can see that we have 3 nodes in Nigeria of H3 Bioinformatics. So this is NABDA in Abuja, University of Ibadan, and Covenant University. And I'll come back to some of their contributions throughout the talk. So H3 Bioinformatics plays an important role in H3 Africa. So the first thing is to ensure that we have computing infrastructure, not just for analyzing data, but moving data. So when we had our first sort of 350 whole genome sequences done in the US, it took up to 3 months to get that data to Africa. And that's just, already a big unnecessary delay. So we really needed to build the infrastructure for moving data. It obviously has to be encrypted because it has to be moved and stored securely. And then, of course, for analyzing the data. So to do that, we've been building tools and containerized workflows. So these workflows allow, it's basically kind of your entire workflow in a black box or a container, and it allows the work to be reproducible. So it's very important in bioinformatics, when you're working with big data, you're going to repeat analyses many times on different data sets to make sure that your science is reproducible and that other people can easily deploy your full set of analysis tools in any computing infrastructure that is accessible to them. So we also provide support for data analysis. We have a big component of standardizing and harmonizing data, preparing data for submission to public repositories, ensuring everything is fair, findable, accessible, interoperable, and reusable. And one of the ways we do this is make the metadata available in a catalog. And then, of course, the huge issue of building skills. In terms of the computing infrastructure, so we've put in infrastructure in several different places. So Covenant University's got quite a good HPC, and they are enabled to do reasonably high throughput data analysis. We also have infrastructure for moving data, so portable hard drives or Globus endpoints. So this is a secure way of moving data. As I've mentioned, we've developed these reproducible workflows. I mean, also assessing capability of nodes to process and analyze data. So we have what we call a node accreditation system, and several of our Nigerian nodes have been accredited in some of these processes. And this is for us to test that if somebody says, I can provide support in GWAS analysis, that we know they actually understand GWAS analysis, they've run the tools, and they know how to interpret the data. We've been developing a number of new tools for human and pathogen data. So we try not to reinvent the wheel. There are so many bioinformatics tools out there already. So we don't want to go and create something that already exists, but there are some that really actually need a bit of adaptation for the nuances of African data, whether it be human or pathogen. And so we have a number of tools that are either developing new algorithms that are appropriate for African data, or a bit about visualization tools for analyzing that data. We also try to maintain software best practices. So we've developed a software best practices document so that these are not just PhD projects and then people move on, that they're bona fide, high quality software tools that match international standards in terms of how they've been developed and the best practices used. So Covenant University, this is one of our nodes, has played a major role in the development of one of these sort of tool suites. So we have they're developing a post-GWAS analysis toolkit. So basically, once you've done a genome-wide association study, or actually any other big sort of high throughput study, you then have a set of tools that you want to run to do pathway analysis, to integrate data, to do meta-analysis, etc. And this is really the downstream biological answer of your research question. And so they've reviewed these tools, they've developed a handbook, and they're providing a nice interface that's provides access to a set of these sort of post high throughput biology analysis. And then we also have some projects that are supporting data standards and harmonization. This is really important when working with big data sets to make sure that the data is well curated and preferably in a standardized way. And this makes it interoperable with foreign data sets. So when you submit to public repositories or when you combine your data with data sets from around the world, it's important that you know that you're talking about the same data element in across these data sets. So we've worked with the Traffic Consortium to harmonize these. We've built standard case report forms for collecting data about different diseases. So this enables people to have their data standardized at the beginning of the project. We also have this data submission center, so it's an archive to ensure that the data coming out of H2 Africa projects is well curated and gets submitted to the public repositories on time. And then, as I said, to make things fair, we then have this accessible via a data biospecimen catalog. So all the data sets and biospecimens that are collected across all the H2 Africa projects, the metadata is collected in this catalog, so you can search and find the data sets or the samples. So that's how we kind of are supporting the data ecosystem for big data in biomedical research. And those are specific examples in H2 Africa. But the biggest, most important component is building the human capacity. If we don't have the human skills on the ground, you lose the data to your international collaborators, and therefore you lose control of those publications, and you lose the value of the research that you're doing. So H3 Bionet undertakes capacity development and training using various different approaches. So we run hackathons and courses. And we've now started, and more intensely with the COVID pandemic, we do a lot of online training, is using a remote classroom model. We do, I talked about the data analysis accreditation project. That is where we accredit a particular node to say, yes, we understand that you know how to do a workflow. We also are working with a carpentries group. So this is, the carpentries provide basic courses that have a very specific way of teaching to make software and data concepts accessible to biologists. So you can have carpentry courses in Linux and R and Python, in genomic data, etc. And so we've been working with the carpentries to build a set of trainers in Estuary Bionet that are qualified carpentries trainers, and then they run courses across Africa. So we've actually got a number of African trainers who are able to roll out these carpentry courses. So they really are a good way of, and only sort of 1 or 2 days, so it doesn't take a lot of time, but it really lowers the barrier to access sort of bioinformatics techniques for people who are not really comfortable using the command line or trying Python programming. So I highly recommend looking at those as when you're doing capacity development as sort of tools for building foundational skills. We also do competency mapping. So we work with the International Society for Computational Biology. So this group has developed, it's an international group, it's developed a set of competencies in bioinformatics. So for a number of different bioinformatics users or audiences, from your scientist who just dabbles a little bit in bioinformatics analysis to your bioinformatics engineer who's developing bioinformatics algorithms, to say what kind of competencies do they need to do their jobs. So this would be things like, well, they need to understand a program, they need to understand how to interpret results, they need some basic statistics, etc. And so this competency work that we're doing is basically enabling groups to say, well, I need just to be able to do, run a few bioinformatics analyses. And then you can see the list of competencies, and those competencies are now being actually mapped to courses. So you'll be able to sort of build up bioinformatics skills using this competency framework. And then finally, we run a train the trainer program. And this is the University of Ibadan node is playing a large part in this project. So we know that, when we started Estuary Bionet 9.5 years ago now, we started our courses having a lot of foreign trainers. And so we would always get trainers from the US, from the UK, and slowly then built up local trainers. So the people who were trained in those initial courses became teaching assistants on the next courses. And then we eventually built into trainers. So we have a formal train-the-trainer program where we try and teach people how to teach, because a lot of people then in your academic setting don't get taught how to teach and how to design courses, how to design a curriculum, and how to deliver that curriculum, especially in the complex case of bioinformatics, where you have multiple different audiences that you're trying to teach, coming in with very different levels of skills, from biology to computer science or maths. And you need to teach the practical application. So bioinformatics is very practical rather than just learning concepts and knowledge. So the Ibadan Train the Trainer program is developing a platform for training trainers. This is the Train the Trainer Advancement resource. And so they've got a number of tools there where they've enabled you to explore bioinformatics topics, find publications, and identify research to build up your curriculum. This is an example of the pages. And so you have various different resources, as I said, and one of these is, for example, to be able to search for bioinformatics topics and then identify sort of content that you could then use for building your curriculum. And they've run a number of different workshops on training trainers. The other group, the other node of Estuary Bionet, NABDA, in Abuja, they have been working, kind of coordinating our project, which is integrating with the faculty training grants. Part of the Traffic Consortium, there are different grants funded by different parts of the NIH, the Wellcome Trust, one of which was the Faculty International funded for training grants to build brand new bioinformatics degrees across Africa. So one of those training grants went to Covenant University, I'll note there, and they used that now to build a new bioinformatics degree program. What we're doing in this project that NABDA's coordinating is actually working with those different groups to say, how do we build curricula that are standard, high quality, providing trainers and excellent examiners, providing research projects. So when you're starting a brand new degree program, often it's because your faculty are not yet trained in bioinformatics. So you need high quality bioinformatics research projects to train postgraduate students. And so what we do in Estuary Bionet is enable research projects to be submitted, have supervisors from the different nodes to start building up the capacity in those different nodes that have not yet kind of got to that level of full faculty in bioinformatics. And then we've also established a joint seminar series with them so that all the students in their program get to present to each other. So they've managed to build these curricula and map these to these ICB competencies. They have identified instructors. So many of our H3 Bionet trainers have now moved to these, gone to these different programs, particularly in Mali, to train and help teach and supervise. And then they've all presented at the seminar series. So that's the involvement of the nodes. So just moving on now to our own capacity development and the models that we use. So we even before the pandemic, we started identifying the need to reach a much wider audience because the demand for bioinformatics training was so high. And so we developed this model several years back that has remote classrooms. So you actually had physical classrooms in multiple different countries. And those classrooms then all joined up online to the teacher and to each other twice a week for 3 to 4 months. And so we had, they would watch pre-recorded lectures, they'd all go online, but in their classrooms, do their practical assignments, join up with the lecturer and talk to each other. So it built a big community of bioinformatics classrooms and managed to reach a wide audience. So you had the experience of being in a physical classroom, but having access to high quality researchers who were doing the training. So the first one we did with that was the Introduction to Bioinformatics online course. This was basic training for bioinformatics users. And so this was basically covering a big set of modules that ran for 3 months, 2 days a week, with contact sessions of about 3 hours. And so there we were able to reach a bigger audience, and now we have over 1,000 participants every year on that course. And as Moses said, they've been hosting a course, one of the classrooms. Unfortunately, now the classrooms are mostly having to be virtual themselves, which makes the logistics even more challenging because you've now got virtual classrooms in a virtual course. We extended this model a couple of years back to run more advanced courses. So we have this intermediate course in a very specialized topic. We've run 16S metagenomic analysis, and then more recently NGS data analysis together with Welcome Connecting Science. And this is a bit more complicated because you need more specialized skills. You actually need to, rather than just point and click web interfaces, practicals use quite complex workflows. So there's more complex infrastructure setup, so you need a higher staff to training ratio, and it's more practically focused. But these have been also again successful in teaching specialized skills to bioinformaticians and bioinformatics users, but across a wide audience and in a larger audience. Another method we use for teaching is hackathons and data jamborees. And so these are more, it's less teaching focused, but you learn. So it's more goals focused. So we say we're running a hackathon to build some workflows, or we're running a hackathon to analyze a particular data set. So you bring a wide group of multidisciplinary people together, they learn from each other, they cross-fertilize, and they solve a particular problem. And so these have been really good at getting goals achieved in a joint sort of manner. So to summarize our training approaches, we used to run face-to-face workshops. We've run more than 30 of these. And actually one of our second big long online, long bioinformatics training courses was actually run at Covenant University in Nigeria. So we had many people from around Africa going there for several weeks. We train the trainer. We've got trainers being developed in the carpentries and in other topics. We also place internships, interns in different institutions. This has stopped now with the lack of travel. We do this live online training, which I've just talked about, and then the hackathon. So through all these training sort of events, we've trained more than 4,500 people. So just to focus on the Nigerian participation. So the Nigerian nodes have contributed trainers. They've hosted face-to-face courses. They've hosted virtual, these remote or virtual classrooms. So this is, for these, it's not only the 3 H3 Bionet nodes in Nigeria, but now this is open to all institutions across Africa. So we've had many additional institutions in Nigeria who are not nodes that have hosted some of these classrooms. And so you can see, and then large participation by trainees. So it's a bit faint here, but on the left is the face-to-face training, so where the attendees are coming from, and you see that next to South Africa, Nigeria is the highest in terms of participation. And when it comes to these online and blended training events, Nigeria is the highest in terms of participation in these events. So we have very good support in terms of trainer and trainees and participation from Nigeria. And then just to finish up with some of the highlights from our Nigerian nodes. So the NABDA node, as I said, they're involved in the poverty training program. They've launched a non-communicable diseases genetic heritage study consortium. So this is to study non-communicable diseases in Nigeria. One of their students, Deborah, was awarded a research training fellowship, Developing Country Scientist Award. One of the graduate students was an intern at Wits University, the Sydney Brenner Institute, and so learned valuable skills to take home. And then Shagun is going to be talking next, I think. He's one of our shining stars and a great example of a success story. So he's recently been awarded an International Intermediate Fellowship on assessing the impact of genetic variation on chronic kidney disease in Africa. This is a Wellcome Trust Award, highly prestigious, highly competitive, and it really is making an enormous impact on bioinformatics across the continent. And then our Covenant University node, this is led by Professor Ezekiel Adibi. They've made active contributions to various projects. As I said, showed the lead, the post-GWAS project. They co-sponsored the first Nigerian Bioinformatics conference in Nigeria, in Lagos State. They started this bioinformatics degree program through this Fogarty International grant, and they've developed various bioinformatics curricula with their computer science department. And then more recently been awarded a Center of Excellence by the World Bank. So again, building of the capacity we've developed and becoming really successful in bioinformatics in Africa. And then finally, the University of Ibadan node, Dr. Angela Makolo is the PI there. They lead this trainer project. They've run several workshops on this, and they've hosted online courses, and they themselves have built themselves up from a reasonably limited node to start with to great capacity now. So that just gives you a flavor for how we've developed bioinformatics capacity across the continent. You can't just think of the human resources. You need to think about the computing infrastructure, the whole data ecosystem, every piece of that. But really the human capacity, if you don't have that, the rest is not possible. So with that, I'd like to acknowledge our funders. So HA Bionet is funded by the NIH, but the Greater Consortium is also funded by the Wellcome Trust. And I'd like to thank the HA Bionet team at CBio and all the work package chairs who really make all these projects a success. And thanks again for your attention and for the invitation to speak.
Thank you so much, Nicola. I'm sure we're clapping. You might not be able to hear it. Okay. Yeah, people are showing their clapping. Okay, cool. That'll be nice. Okay, so I just want to ask the house if there's any question. I'm just going to entertain two. I see Shagun is already ready, and we're going to go to him quickly. So two questions quickly. I don't see anyone on the chat. Somebody hands is up. Is that Toyin Ademola? Toyin Ademola, your hand is up. Can you go ahead, please? You can unmute yourself. If your hand is up for the question, Toyin. Is there any other person? I can't see anyone else. Can Toyin hear me?
I think he cannot unmute.
Oh, for some reason.
Unless if the host does that. So, Aniedu, could you unmute Toyin Ademola?
Can you? Okay, I see. I can ask him to unmute here too.
Akinola is also.
Okay, so I'm just going to take those two, please. Tony, we just ask you to unmute. Try it again, please.
I think his question is already also on the chat, so you can.
I already listed my question.
Oh, okay. Oh, okay. Beautiful. Okay. So thank you, Mr. Tony. So his question is: these HPCs, I guess, like the one you talked about, located at the Covenant University, are they available to researchers outside the H3 Africa nodes? That's his first question. Then the second question is about the data access. Are these H3 Africa data repositories you spoke about also available to researchers outside the node? Okay. Nicola, do you want to answer this now, or do you want to take it together?
Yeah, sure. I can answer those now.
Okay, cool.
So thanks for the questions. So we so for Covenant University, some of the HBC already existed. And so their computer science department will have, regulations about who has access to them. But where we have supplemented that with H3ABINET funded equipment, then we have an expectation that that equipment should be made available to researchers in Africa. So certainly at UCT, we've, put money into a big cluster. And the idea of that cluster is that anybody who's doing bioinformatics that has a sort of a bona fide research question can access that. So it's best to contact Professor Adibi, and but the idea is that it should be available to to other researchers in Nigeria and the region. On the second question, yes, H3Africa data gets submitted to the EGA, the European Genome Phenome Archive, and all access to anybody around the world is controlled by a data and biospecimen access committee. So you go into our catalog, you look for the data sets, and then you request access. So everybody can have access no matter where you are in the world. If you're an African researcher, you can also approach the H3Africa PIs directly. If you see some data sets and you're interested in them, then approach the PIs, because building African partnerships is one of the aims of the H3Africa Consortium. So those kinds of things are encouraged.
Thank you, Nicola. And I think one of the things that the organizers can do is to post the link to some of this data repositories so that people can take home from here. Okay, Akinola, please. Akinola, your hand is up, or do you want to type it? Or you've typed it? I don't see you not speaking.
Maybe he can't unmute. There's another question in the chat about
please go ahead.
Basic mathematics. That's a difficult question, actually, because it depends on the background. So we found that, and it depends where you want to go to. So people say, I want to know how to do bioinformatics, but bioinformatics is a huge subject. So really, it's actually that you want to be able to do bioinformatics for a particular research question normally. And so what we suggest is that you gain foundational skills in data-related things like Linux, command line, a little bit of Python, some basic statistics. And that shouldn't take you more than a few months to do a few courses, but have real data to work on. If you do courses and then you go away and then you do some more courses, unless you've applied it, the skills don't really sort of stick. So you can do those in a few months and then do a specialized course on a particular topic area. If you try to be a jack of all trades, well, then it's going to take you forever, because you've got to learn each new topic research question or each new data type is quite complex. And so you really need to go into depth to become really competent in that. So pick the one or two areas that you want to become proficient in, get the basic foundational skills, and then dive into those areas. But always make sure you've got a research question to apply your skills to immediately, because it cements the skills, and then immediately you see where the gaps are in your skills. But a few months, and again, it depends on your background. So if you have absolutely no computing capabilities whatsoever, it might take a bit longer. But most of the youth today have doubled quite a lot in devices, and so I would just say a few months, and then to become more specialized, maybe the rest of the year, and a lot of practice.
Thank you, Nicola. All right, so that's quite insightful. I remember at the beginning I was a bit confused as to where to go. I was checking Nicola out what she does. I also remember checking Shegun out what he does. What he was doing at the time. Eventually, I decided on. So bioinformatics is a tool that you need to apply to a specific research question. Akinola, sorry, I don't think I can entertain your question for now. We'll have to move on. Okay. Once again, we appreciate Nicola for that opportunity and that insightful presentation. And once again, we appreciate your time to spend. Okay. Thank you so
I think there are some questions on the chat, and Nicola still has like 5 minutes left.
Do you say, okay? I'm advised that she still has 5 minutes left, so we can go ahead.
Yeah. Akinola have already typed his question.
Okay. How to work with H3A Bionet, I believe.
Yeah. for those who are outside the shore
Outside
of
the shores
Africa.
Yeah. Can you see that, Nicola?
Yeah. So we work with people around the world. So we work with them in many ways. We work with the global bioinformatics training community. When it comes to researchers, we do have collaborations with people all around the world, especially expats, people who are from Africa. What we try to do is we try to establish what is the nature of the collaboration. Is it something that you want to contribute and to help build capacity in Africa, or is it that you want to come in as an equal collaborator? Or is it, what is the interest in the collaboration? But we're completely open to working, and we do work with many people around the world. So there is no kind of limit when it comes to capacity development. Obviously, our focus is on African countries, and also for time zone issues, when we're training people. But when it comes to research collaborations, building tools together, building resources together, we open to collaborations with people around the world, and completely welcome that, because you bring in a new perspective and resources in usually the higher income countries that bring in resources that we don't have in Africa. And so these kinds of cross Atlantic, cross English Channel borders collaborations are very much welcomed.
Cool. Thank you so much. Okay, I think we can still take one or two more. If she has 5 minutes. Anybody want to type quickly?
Yeah, there's one before Akinola.
Okay, let me go for that.
Kufra Abasi Abanekot.
Kufra.
Yeah.
Okay, he was asking about the train-the-trainer program and the workshops. Are there for specific target persons? And what time in the year does it come up? It's just a schedule.
So our training program is quite diverse, and it's become so busy we've had to kind of slot everything in. So the trainer is for people who are going to be running courses themselves, or going to be in faculty positions to learn how to teach. So there's some that are online, and so they run sort of ad hoc. We're planning to run one with the Wellcome Trust in about the first quarter of next year. The IBT courses are targeted to non-bioinformaticians, so they're non-specialists, so it's researchers, bioinformatics users who want to do some bioinformatics for their research. Those happen, so we've just finished one now, so those start in about July. We're running a next generation sequence course, so a specialized course in NGS, probably starting around March next year. And then our intermediate metagenomics course usually runs after, or just slightly overlapping with IBT. So that's sort of, was also just finished actually. So that ran over the last few months. So we have a website, and the website has the list of upcoming courses, and each course clearly defines who the target audience is. So try to go there, see who what kind of course you're looking for, whether that course suits your background, and then it'll give information on the course that's run. If you missed a course, don't worry, because all our materials are recorded, they're placed on YouTube, they're made completely openly available. So you can also follow the course in your own time afterwards if you happen to miss it. But a lot of these courses we run annually. So, there's usually an opportunity the following year to join them.
Okay, maybe I have a question for Nicole. I would say thank you so much for your presentation, and also the opportunity you have also given to the consortium to host the IBT for 2019 and also 2020 and 2021. Now, you could observe that we have 3 nodes in Nigeria for the H3 Bionet, 2 are in the West, and 1 is in the North Central also. Yeah, so are there plans to increase these nodes across the country, because Nigeria is quite huge, and sometimes when they have a limited number probably to admit in a training, maybe they give priority the people they know or people that are around them, so this does not cut across the entire country. So so what do you think, and how do you plan to address this? Because Nigeria is quite huge.
Yeah, so it's a tricky one. So H3 Bionet, people want to keep wanting to join H3 Bionet, but it's a funded project. They had a defined budget for 5 years, and in fact, it's coming to an end next year, possibly with a short extension. So H3 Bionet, as we know it, that funding will come to an end, and that was defined. So we defined budget. So we had the groups that had joined from the beginning. But we do have other initiatives. So we're trying to get other groups involved by, having classrooms, building communities. And the African Society for Bioinformatics and Computational Biology is another way to build, this community of bioinformatics institutes around the continent. As a sustainability measure, we also investigating the idea of a more Pan-African Bioinformatics Institute. And so we're about to actually put out a survey. We're developing a survey that'll go out, and so I'll make sure it gets to your groups. It'll see who is doing bioinformatics on the continent, because it's changed a lot. The landscape's changed enormously since we started, 9.5 years ago, to see what is the current landscape, where is their capacity, and how we build this sort of more Pan-African, more inclusive African bioinformatics community. Not all will necessarily be, part of an institute, in a formal institute, but we hope that as many groups as possible can get involved in these activities, whether they be training, research collaborations, etc. So those are the plans. I mean, Eshri-Bandit, as I said, has always been restricted by how much funding we have and our massive set of deliverables that we've had to get done. But through the society and through this potential future institute, I think there will be scope to move beyond human genetics, agriculture, other applications of bioinformatics. So there will be opportunities in the future to build wider networks.
Thank you so much, Nicola.
Thank you, Nicola. Once again, we appreciate. Thank you so much for the presentation, and I believe those have been insightful responses to the questions. Thank you so much. Okay. Once again, we appreciate you, and we'll if you are with us till the end, fine. If not, we wish you well. Okay, we move on, and we're going to have a presentation by another important personnel, Dr. Segun. Let me correct myself, Professor Segun Fatumo. He's an associate professor of genetic epidemiology and bioinformatics at the London School of Hygiene and Tropical Medicine, and is a group leader at the African Computational Genomics Research Group that is located at the Medical Research Council and the Ugandan Virus Research Institute in Uganda. Dr. Fatumo research likely focus on genetic impact of non-communicable diseases in Africa, with particular interest in assessing the impact of genetic variations on kidney function, diabetes, lipid metabolism, and a whole range of cardiovascular and cardiometabolic traits. And I'm sure that's very relevant to Africa. He's actively involved in teaching and PhD supervision across different universities in Africa, including Makerere University in Uganda, University of Sciences, Techniques, and Technology of Bamako, Mali, and Covenant University in Nigeria. He's a fellow of the Higher Education Academic in UK and teaches on an MSc program at the London School of Hygiene and Tropical Medicine and Queen's Mary University in London. Dr. Fatumo has been featured in notable media outlets, including the Science magazine, the coveted Forbes magazine, the London School of Health and Tropical Medicine Press, ScienceDirect.net, African Vibes, Research Professionals, etc. He gives regular invite and keynote presentation at major conferences around the world, around the globe, reviews for many high-impact journals, including serving as associate editor and member of the editorial board. Dr. Fatumo received postdoctoral training in genetic epidemiology at the University of Cambridge and Wellcome Sanger Institute, and a postdoctoral fellowship in bioinformatics at the University of Georgia, Athens in the US. Prior to that, he had a postgraduate training in applied bioinformatics at the University of Cologne in Germany, and a PhD in computer science with a focus on bioinformatics from the Covenant University in Nigeria. Dr. Fatumo is also the founder and the pioneer president of the Nigerian Bioinformatics and Genomic Network. He is the immediate past vice president of the African Society for Bioinformatics and Computational Biology. I wanted to welcome Professor Segun Fatumo as he presents to us. You're welcome, sir.
Thank you very much for the introduction. I would like to start by just acknowledging Nikki Moda, who has just given an excellent presentation. Everybody know that without the work that you did, the foundational work that you did, some of us will not be anywhere today. So I'd like to thank you so much for those work, many years that you have committed to bioinformatics in Africa, which all of us are enjoying today. Thank you very much, Nikki. So good morning, everybody. My name is Shegun Fatumo, and today I'll be making this presentation, which I applied to developing African bioinformatics and genomics for novel gene discovery and precision medicine. As already introduced, I'm an associate professor at the Department of Non-Communicable Disease at LSTCM. I lead a group called the African Computational Genomics Group at the MRC UVRI in Uganda. So I would like to also recognize the Vice Chancellor of this university, who I understand has given a presentation earlier, and as well as all the management of the university. I also like to acknowledge the current president of the Nigerian Bioinformatics and Genomics Network, Dr. Charles Aditoji, and also the members of MPGN that are here. I would like to say thank you very much for the executive of this consortium, and also for inviting me. So if you have any question that I'm not able to answer today, please feel free to follow me on Twitter. Here's my Twitter, and feel free to send me an email if you have to. Of a lot of genomic data.
Can you hear me?
So genomic data, we know that. So African level of diversity. Oh, can you hear me? Can you hear me?
Yeah, we can hear you now. Go ahead.
We can hear you now.
Yeah. So genomic data are key for precision medicine, and African data are vital due to its level of diversity. African genomic data are very scarce, and you know that. Very, very scarce in continental Africa. So I always like to differentiate between African data, because there are African Americans and Africa in other places, and continental Africa. So African data are scarce, but Africa data from the continental Africa are very scarce. So I'd like to mention that African scientists with the skill sets to analyze large-scale genomic data are scarce. And you can see from what Nikki was presenting today, all the effort is to develop that skill set for people in Africa. Very scarce in Nigeria, relative to our population and also cultural diversity. So we need people that have the capacity to be able to analyze large-scale genomic data in Nigeria. So in this presentation, I intend to show how we are using genomic resources and also bioinformatics capacity building in Uganda for novel gene discovery and genetic risk prediction. So my aim is that the model that we use in Uganda might be useful for people that potentially plan to develop capacity in bioinformatics and genomics in Nigeria and in Africa generally. So in Uganda, we started by setting up the genomic epidemiology study of non-communicable disease by building what is called general population cohorts that have been existing since 1989. So this GPC is an annual program where we go to a community and do sample collection, both urine collection, blood collection, and also do other study focusing on non-communicable disease and communicable disease. In 2011, precisely, so this research activities was extended to do genetic epidemiology of NCDs in Uganda. So every year, we support this team by having an engagement with the team and with the community, starting with very strong community engagement with the stakeholder, doing the annual survey, and also using the resources that we have from here to do our study. So in Uganda also, we have the high-performing computing, which is one of the best in Africa currently. So what we have in Uganda is about 20% of what you will see at the Wellcome Sanghan Institute. So we have the biorepository at the MRC UVRI in Uganda, where the blood samples that are collected in the community are stored. So one of the things we are doing also is collaborate with other African scientists in Africa. You know that at the beginning I mentioned that the African data sets are very, very scarce. So what we do is that collaborating with other cohorts. There's a cohort called ADAM. This cohort is a collection of samples from Nigeria, from Kenya and Ghana. So there's a cohort called Durban Diabetes Study in South Africa, a cohort called Diabetes Case Study also in South Africa, and also a cohort from Uganda. So we are leveraging a lot on collaborating with other studies in Africa. I think this is key for any bioinformatics or genomics that will be successful in the continental Africa. So we collect a range of phenotype from lipid traits, blood pressure, red blood cell, platelets, liver function, and so on and so forth. And we study this. In 2019, we published the largest ever genomic study of Africa because we are leveraging on this collaboration with other people in Africa. So this study appeared in Cell. Is currently still the biggest study of genomic in Africa, and I believe that other will be coming on later. So in this study, we identified 10 novel associations associated with various traits. Here you see a BMI, HbA1c, and also red blood cell, and also other liver function traits. So these are key because in study in the European population, where they have sampled even up to 1 million people, this association were not found. And the reason is because there is a lot of genetic diversity in African population. And so this SNP, for example, that we found to be novel and associated with various cardiometabolic traits are not found in those European population because they're not present in those population. So again, this emphasized the need to study African data set and study large-scale data set from Africa. So now we are now extending our collaboration, now trying to aggregate data from MVP. MVP is a Million Veteran Program. It's a program that was established in the US, and they are collecting samples from about 1 million people, but they have, fortunately, about 57 African Americans. So we are collaborating in MVP. I've mentioned the 14,000 from different African ADAM, DDC, and DDS, and also the GPC collection. And also we are also leveraging on the UK Biobank. So UK Biobank also is a program that is collecting 500,000 people from the UK. Fortunately, they have 7,000 people of Black participants. So I'm emphasizing this because this collaboration is very key to have an established bioinformatics and genomics in Africa, and specifically for Nigeria. So what we are doing is to now build capacity with our level. For example, here in the picture here is Brenda Udoze, who is a Nigerian, but working in my group. She did a master's in bioinformatics at the University of Science and Technology. So what she's doing is she's analyzing the largest meta-analysis and multivariate GWAS or blood pressure in Africa. If you are familiar with a GWAS study, the largest GWAS of blood pressure you will ever have seen will probably be about 10,000. But Brenda is now analyzing the largest ever using leveraging on other collaboration. You can see here she found also a novel gene that had not been found before because sample size were small using a different method, both meta-analysis and multivariant study. So here again is Ture. Ture also is an MSc bioinformatics study at the University of Science and Technology. So what Ture is doing is using an approach called file mapping. Different file mapping approach. There's a multi-trait file mapping and also single trait file mapping. Trying to see what is best with within the African data set. So in the group, if you are familiar with an approach called polygenic risk score, so all these, the genomics and also bioinformatics, we are trying to see, are we able to group people and also predict if some people are going to be having some kind of disease in the future. So I think this is a very great approach, because if you're able to identify people that will have, say, type 2 diabetes in 5 years and 10 years, so it will help the government to provide necessary intervention to support them. So we in the group we are working on doing PRS of lipid trade and also PRS of type 2 diabetes, and leveraging on the data set that we have in Africa. So previously, if you're familiar with PRS, you'll have seen this kind of picture in the opposite direction. You'll have seen that the PRS is not predicting very well in African population. But here, because we have access to several data sets from African ancestry individuals, we are able to predict lipid trade in African population much more better. And so this is what I was talking about PRS that is very common. This is from a paper by Alicia Martins that showed that PRS doesn't predict very well into African population. So I think one of the reasons for this was because we don't have an appropriate data set. But in our group, we are aggregating data sets from different African populations, and we're able to produce PRS better using African ancestry individual data set. So this is a paper that was recently accepted in Diabetes Care. This was led by me and my colleague at Nashe. So here again, we're showing that we're able to predict PRS. Here you can see the African population, African replication. This is also from Adam, from Nigeria, Kenya, and Ghana, and European population. And same story is that we're able to predict type 2 diabetes from African ancestry individual into different African population much better than other data sets. And here, this is Chizom Shuremeko, also from Nigeria, in my group. She is studying, doing a PhD in Makerere University in Uganda, and she's trying to put together different data sets from type 2 diabetes and other related type 2 diabetes data to find an association, and also to predict a subgroup of people that might have a particular disease in the future. So this is a pilot work that she did with a collaborator in South Africa earlier this year. So this is a paper we published this year showing assessments of metabolic risk and the risk of stroke in individual of African ancestry. So here we showed that this one is African, the color look like yellow, and the European in blue. Here we show the risk of different traits, including cholesterol, LDL, and HDL. And you can see here, increased risk of stroke with HDL and LDL, and HDL showing protective for stroke in African ancestry individuals. And this work was recently published in a journal called Stroke. So again, using the data set that we have in the group, recently we published the first ever GWAS of kidney function in Uganda population and as well as African population. And we identified 2 loci, and they also fine mapped those 2 loci. And this work was published in a journal called Human Molecular Genetics just earlier this year. And this is very important. This study is under power. So we're now working with other people across Africa to aggregate kidney function GWAS data set and be able to discover what we're not able to discover due to low power from this study. So Richard Mayanga, who is a second-year PhD in my group, what he's doing is to now aggregate data from DDS from South Africa, so Adam from 3 populations, Uganda resource, which I've earlier mentioned and which we have published, the Awujen from Michelle Ramsey group also in South Africa, and also a new cohort called Meru. So this is a cohort from Malawi, and we are currently genotyping up to 8,000 people here. So Mayanga is now building capacity to be able to do quality control, imputation, GWAS, and as well as meta-analyze all these core together to be able to have power for gene discovery and also for precision medicine. So recently in a pilot that was done by Mayanga, so because these 2 data are currently available, So what he has done is to meta-analyze these two data sets together. And already we find an association with a gene called PDZK1. And fortunately, we saw a very strong expression of this gene in liver, kidney, and liver function. So this is a very promising gene that was found by Mayanga. But before we validate this, so what Mayanga will be doing is now to replicate this this association that we find in other population in Africa. So as part of our investigation to understand kidney function in African population, so Mariam, who is also from Uganda, she's a research fellow in my group, is working to determine the frequency of two APOE1 risk allele, that's G1 and G2 in Uganda genome resource that we have. And she's trying to investigate the association with kidney function, blood pressure, and bilirubin level. So this slide, for example, shows the high frequency of one or two copies of APOE1 risk allele in Western African country, in Nigeria, Gambia, Sierra Leone, than what you will see in Eastern and Southern Africa, in Kenya, Uganda, and South Africa. So this is also consistent with the prevalence of kidney disease. What that means is that where you see high prevalence of kidney disease, you also see high frequency of one or two copies of this risk allele. So we think this is a good pattern, and Mariam is now looking to write up this for publication. So in our recently published work on GWAS of kidney function in Uganda population, so we reported that multi-ethnic based PRS do not predict well in the Uganda population. So this is now Christopher Kitu, another second year PhD student at Makerere University. He is now developing a polygenic risk score for kidney function from diverse population and evaluating the transferability of the kidney function for kidney function into different ethnic groups in African population. So the genetic risk score developed can be used as instruments to assess whether cardiometabolic traits or disease are causally related to risk of chronic disease in Africa. So this is Dr. Okoyemi Shuremekun, who is a postdoc in my group, and is now leading a bidirectional two-sample Mendelian randomization study using EGFR as both exposure and outcome to investigate the causal relationship between each cardiometabolic trait and kidney function. So I brought all this together because these are important to see how we are building capacity and how we think that building capacity is key to have a strong bioinformatics and genomic platform in a country like ours in Nigeria. So now and the future, so my proposal is that we now need to start thinking about public-private partnership to be able to have a stronger landscape of bioinformatics and genomics in Nigeria. So about 2 years ago, so we established NCDJHS, which is a consortium that is partnering with a private organization in Nigeria. So on the left here is Dr. Abasi Eniobong, who is the CEO of 54 Gene, a biotech company in Nigeria. And next to him is Professor Babatunde Salako, who is the Director General of NIMA in Lagos, a public organization. Here is a lecturer at the University of Lagos, Dr. Omolola Salako. This is myself, and also Professor Oyekomi Nash, who is also a director at NABDA. So we are putting together the partnership between public and private establishments to now move forward and do the largest, what we call the Nigerian 100,000 genome project. So, so the aim of the project is a nationwide genetic epidemiology study of non-communicable disease in 100,000 people. And currently we are collecting samples across Nigeria, everywhere in Nigeria, there's 6 geopolitical zones. So we have collected samples from 14 states, 23 sites, and involve so many professors in all those sites, both in the hospital and also in the community. So NCDJHS, for example, we have 2 projects. There's one that's focusing mainly on collecting samples from from the hospital, and the Nigerian Cardiometabolic Genome Study, called NICGEN, is collecting samples from the population. So as I end, so again, the Southeast Bioinformatics Symposium, I want to thank you again for organizing this meeting. I can see from the team that you are looking forward for a way forward for bioinformatics in the Nigerian landscape. So here are my submission, and you can see that clearly from my presentation. My submission is that to get a way forward, we need collaboration, very strong collaboration, strategic collaboration. We have to be consistent. Over the years, since I started bioinformatics, I've seen people come and they go, start something and they leave. So we need to be consistent, and we need to focus on capacity building. Earlier this year, a group of students asked me to talk about my contribution to bioinformatics and genomics, and how they can leverage all my own experience for their own as they are starting their own career. So this was the slide I used then, and I would like to end with this slide about my own contribution to bioinformatics, what I've done in bioinformatics and genomics, particularly as it relates to Nigeria. So in 2014, I had the word bioinformatics for the first time, and I know some people also will be hearing bioinformatics for the first time at some point in their life. In 2016, so I became the first person to earn a master's degree with bioinformatics specialization in Nigeria, and that was in Covenant University. In 2017, so I founded the ICB RSG Africa and led over 500,000 students across Africa. That happened in Kenya, and Nikki Moda was there in 2017 where we started the student group. In 2019, so I discovered 20 potential drug targets against malaria, some of which have been validated experimentally, and I became the first person to have a PhD with bioinformatics specialization in Nigeria. In 2012, so I decided to use my bioinformatics skill on large-scale genomic data set, and started another postdoc in genetic epidemiology at the University of Cambridge and Sanger Institute. 2013, so I co-led the current largest ever genomic study of African and published in Cell as co-first author. In 2014, so I led the publication on bioinformatics and genomic bioinformatics and computational biology in Nigeria that was published in PLOS Computational Biology. So if you want to know about the story about bioinformatics in Nigeria, so we put everything here in 2014, and I think that is now due for update. In 2018, so I founded and served as the pioneer president of Nigerian Bioinformatics and Genomic Network, and I handed over this organization with over 2,000 members and well-established network earlier this year. In 2019, so I led and co-organized the first ever bioinformatics conference in Nigeria. In 2020, I established the African Computational Genomics Research Group at the Numeracy Uganda and LS Station. In 2020, I also am co-leading the NCDJHS. That's a very giant project that is aiming to recruit 100,000 Nigerians for genomic study. In 2020, like Nikki mentioned, I won a prestigious grant award of over $1 million US and higher African PhD and postdoc fellow. In 2021, I published the first ever GWAS of kidney function in continental Africa. 2025, like I told the students, I aim to restart much stronger. I become a better person. So that's the story of my bioinformatics. I would like to thank members of my group. Currently, we have a postdoc. We have 4 PhD students, 3 of them in Makerere University in Uganda, and 1 at London School. And we have also a research fellow. So many people to acknowledge for amazing collaboration that has helped with my own bioinformatics and career network, particularly at the HU Bionet, Nikki Moda, Oyekominash, Azikiwe, and others, and also other people from my postdoc and currently at the Numeracy Uganda. Just before I go, in my group, we are now currently looking to hire a senior postdoctoral fellow who has capacity to do GWAS, fine mapping, PRS, Nigerian administration. We look for a person that will be able to live in Uganda or Malawi. So if you are interested, please feel free to contact me. So thank you very much, and I'm very happy to take questions.
Thank you very much. Once again, we indicate our clap for that wonderful presentation and highlighting all that you have done for bioinformatics in Africa. So please, the floor is open for question. I already have a question on the chat for you. I also have a question, but let me start with the one I have on the chat. So Professor Segun, there's one on the chat says, are there any plans in your group or others that you're aware of to integrate this data with other omics data, which referring to the transcriptomics and the proteomics data, in order to drive our mechanistic studies in Africa? Thank you. Do you want to take that, or do you want to take a couple of questions?
Yeah, I can take that. So currently, we are trying to be very strategic. There's so many research questions to answer, and we understand that we don't have the capacity to answer all the research questions. So currently, and up to the next 3 years, our focus is going to be mainly on genomics. So we're going to be using those methods I mentioned, GWAS, PRS, and data randomization, and file mapping. So when we see that we are well established in that, we are also already establishing collaboration with colleagues in Germany to be doing proteomics and also be doing metabolite profiling. But we are taking this step by step. Yeah,
very, very good. I like the emphasis on focus and the capacity, using the capacity you currently have to do that. Okay. Somebody wants your email. The organizers can give for the person. Okay, there's another one on the chat. He's asking, please, what's the structure for the recruitment of the project you spoke about, recruiting 1,000 Nigerians for the genomic research?
So we have the 100,000 genomic project that is currently being recruited. And I think this is a good opportunity for people that have the skills to be able to analyze this data. So in the past, what we have seen is that you see sample collected in Africa, and those samples are now shipped to Western country in the US or in the UK for others to analyze. But that has changed. Now we have some capacity, but I personally think that we don't have enough capacity to do a landscape genomic study, particularly in Nigeria, now that we are collecting 100,000 samples across Nigeria. So I think people that have the capacity will have seen a lot of advertisements from SJHS, G4Gene, asking for people that can do this analysis, but it's always very, very hard. So one thing I will really love this symposium to achieve is if you can build that confidence in people to really build capacity. You know, one thing I've seen is that many people do like to attend any workshop, any training without being very focused on what they actually want to do. But if we have people that are very focused and they build necessary capacity, necessary skills to be able to analyze this data, I know that the sky is going to be the limit. So I think there are a lot of opportunity there to analyze this data, but there are not a lot of people that have the necessary skill set to analyze them.
Okay, thank you. I think you've answered one of the questions on the chat. One person was asking the involvement of Nigerians bioinformaticians in that particular project. I think you've alluded to the fact that the capacity might not be there correctly, but I'm sure that if there are capacity on the continent, particularly in Nigeria, you will use them to analyze that. So the same person is also asking, is there currently in your group a focus on drug discovery? Do you intend to go that in the nearest future?
Yes, the ultimate aim of everything that we do is drug discovery. And if you are familiar with my project, my work, you'll have seen some paper published on drug discovery. So on the question of Nigerian being involved in that 100,000 genome project, the answer is yes. So we recently hired a postdoc for this project. And we have also recently about to award 4 PhD scholarships for people also to work on this project. So there are opportunities for people to work on this project. I think if you have a look at the website that capture NCDGHS, you will see that opportunity. So for this year, people that want to do PhD, we've already done and awarded this scholarship for them. This will be every year ongoing. If anybody is interested, in doing and building capacity in genomics analysis, that is an opportunity to do so.
Okay. So 2 more questions I will just take before we move on. So there's I think I echo these questions. Are you in collaboration with any genomic for agriculture organization, or are you aware of any opportunity for that? I guess this person is more interested in, can we adapt this platform that you have to sample, plants and the rest of them to agriculture?
Okay, like I said at the beginning, there are so many very interesting research questions. And so my research group, we are focusing on the one that we have the capacity to do. And our focus has been human genomics. I've seen a lot of work on agricultural genomics, which are also very interesting. There are very relevant people that can help. Maybe I don't know whether Thank God is here, who is interested in agricultural genomics. Maybe that question can be addressed to him.
Yeah, thank you so much. So 2 more, I'll just read 2 more quickly so that you can just take them. So somebody is interested in your MSc program. If there is sponsored MSc program, I think you alluded to that already. There's another question, I'm just trying to find it on, related in terms of MSc mentorship opportunity for them in your group.
So the student I currently supervise for MSc, they are sponsored on one of the programs at the University of in Mali, and also there are also opportunity for I don't know whether Nikki mentioned about data science. So currently, our collaborator at the Macquarie University also was awarded the data science training program. So there's opportunity also to do some genomic data analysis with that. There are opportunity for sponsorship for that. So I think if anybody wants anything, you just will look for them. There are opportunity everywhere. On my group website, we always post opportunity for postdoc or PhD opportunity that we have. Just be on the lookout of that. If you're also on Twitter and follow History Bionet or History Africa, you will see a load of opportunity for PhD scholarship or PhD fellowship or MSc fellowship.
Okay. Yeah, I have a question I just want to ask before you go. And someone is also asking about your contribution to training researchers in Nigeria. I think you alluded to that in your presentation.
Yeah.
I don't want to put
that. Everything I do is training. I currently have 3 students I supervise for PhD at Macquarie University. One of them is Nigerian. I do training in Nigeria. I'm going to Nigeria again to do training. So everything I do is training, and everything is focusing mainly on Africa. So I do training. I don't do all training. My training is focusing mainly on genetic epidemiology, GWAS, polygenic risk score, Mendelian randomization. So there are a lot of opportunities for training. And if you were here at the beginning when Nikki was presenting, you'll have seen also loads of opportunities for training.
So I think what the organizers can do is, with the permission of the presenters, you can make the slides available. And I believe there will be some sort of summary at the end of this, where we can attend to those scholarship opportunities. So please, that would be all for that. So my question is, in terms of some of the publications you've made, you have the metadata available for these databases. Is there access control for that? And secondly, you spoke about public-private partnership, and AWX, Amazon, and the rest of them have a very good platform in terms of cloud computing. What has been your experience in engaging such large organization in order to access, high-performance computing?
Okay, so we have our data available on EGA. So EGA is where you store your data, where you publish, and we've had people request for that all over the world. We are always very happy to share our data with people. So if you are familiar with EGA, you will have seen that a process of applying for those data, they are available. So on a public-private partnership, see, this is an opportunity that when you are rightly placed and your work is being prominent, people are going to invite you for many public-private partnerships. I think if I stay students, one best way to start is to engage by doing PhD or doing postdoc, and let your work be there in terms of publication. and the opportunity for public-private partnership will be there. Or if you're looking for opportunities to work in an industry, maybe if it's forging or also a very similar organization might be appropriate for you to have some experience with internship.
Okay, thank you so much. I believe that we have been really, really informed, and we are blessed by your presentation. And I am particularly happy to see you close up, even though virtually. And I really appreciate your time. Once again, we thank you, and I'm sure everybody on the platform appreciate your time. Thank you so much. If you're still on the platform, thank you. God bless you.
Samuel, what is the issue? I think we have to, as we wait for the chair of the technical section, maybe to ask, thank God, Ebenezer already, if he's here.
Sorry, I was out.
Okay. Okay.
All right. All right. So the next presentation will be by Thank God Ebenezer.
So you need to make Thank God as Ebenezer co-host.
He'll be presenting on the African Biogenome Project, genomics in the service of conservation in Africa. Dr. Thank God is a bioinformatician at the European Bioinformatics Institute in the UK. Within the UniProt team, his role focuses on developing computational pipeline for automatic functional notation for proteins using evolutionary models. Prior to joining EMBL-EBI, Dr. Ebenezer was a postdoctoral researcher at the Hellham Institute, a UK national facility for genomics, and where he worked as a wheat transcriptomics and epigenomics. Dr. Ebenezer studied for his PhD degree in biochemistry at the University of Cambridge, UK, where he worked on the genome of Euglena gracilis. Euglena gracilis. He has an MSc in environmental toxicology and pollution management from the University of Lagos, Nigeria, and a BS in parasitology and entomology from the Nnamdi Azikiwe University, Awka, Nigeria. Aside from dry lab science, Dr. Ebenezer is involved in fostering dynamic international, regional, and local collaboration in science. Recently, Dr. Ebenezer initiated the formation of the Euglena International Network and the African Biogenome Project. Dr. Ebenezer also instrumental in the initiation of the current platform that we are on, the NAU Genomics and Bioinformatics Concept. Let's make welcome Dr. Thank God Ebenezer for his presentation.
Thank you, Dr. Samuel Gai, and thanks to Moses and the organizing committee for inviting me. So I'm just going to share my screen. This is likely to be a slight shift from maybe the presentations that have been made. So I'm trying to just share my screen. Now I can't find. Okay, I think I can find it now. All right. Okay, can you see my screen, please?
Yes, we can.
Okay. So thank you once again for inviting me, Moses, and the organizing committee. My name is Thank God Ebenezer. and I'm a bioinformatician at the European Bioinformatics Institute. And today I'm going to be talking to us about the African Biogenome Project, basically which focuses on sequencing genomes across the continent of Africa, mainly within the non-human space, plants, animals, microbes, just about any specific genome. And I'm also the co-chair of the project. So I'm going to be talking to us about providing a bit of a background to this and talk about the delivery pipeline and the organization of the project. So this is also similar to the project, the presentation I made at the Nigerian Bioinformatics conference, only because the project is evolving and we need more African scientists to get to know about this ambitious project. So if you attended it, there's a chance that there will be some slight variations, but it's heavily similar. So before I go into that, I'm just going to talk briefly about myself. And I grew up in Nigeria, and I did my bachelor's in parasitology at Nnamdi Azikiwe University. Then I went to the University of Lagos for my master's in pollution, environmental toxicology and pollution management. And I did my NYC in Kefi. Then I worked with Shell for about 3 years. And then it was at Shell that I found the need to pursue a PhD at Cambridge. So I worked on the genomics of Euglena gracilis. And then I worked at the University of Oxford. I worked at the Wellcome Center for Infectious Diseases at the University of Dundee. Then I went to the Airline Institute for my postdoc, and then I am now at the ABI as a bioinformatician. So I am involved in a couple of things, but I'm just going to focus on the African Biogenome Project, which I think is more relevant to the discussion here today. So, and basically the two major ones are the African Biogenome Project and the Euglena International Network. Mainly, the Biogenome Project mainly was something I started thinking about a few years ago. And when I was at a postdoc at the Airline Institute, I put together the first proposal towards it. And also the Euglena International Network was more from my PhD. So basically, I also think that in order for us to ensure genomics for all, have some diversity in genomics, we must and should build local communities at the national level, at the regional levels, basically. So in 2018, I joined efforts with Dr. Shegun Fatima to set up the Nigerian Bioinformatics Network. And I also helped to initiate the NAU Genomics and Bioinformatics Consortium through Professor Osi Nwago and also Moses. So today I would focus on the African Biogenome Project. So what are the goals of the African Biogenome Project? The primary goal of the African Biogenome Project, which is the first goal, is mainly to coordinate all non-human sequencing efforts across the continent of Africa and act as a forum for the non-human space. And the second goal is to initiate, develop, and support new non-human genome sequencing efforts, as well as sequence over 100,000 species across the continent of Africa. Then the third goal is to use the data to actually contribute to regional biobanks, Africa CDC platforms, the AU, the Africa Union Inter-Africa Bureau for Animal Resources, and just about any continental agency that we believe would or should actually start using this data. So just a bit of a background to the project. We believe that, for instance, that the global biodiversity landscape is a shared heritage, and nearly all continents is endowed with a form of biodiversity. And there is a 6th mass extinction event currently happening, which require us to urgently begin to turn the tide. So there are about 52% of the earth-based mammal fish under extinction. And this also includes about 39% land-based species and 76% freshwater species. And the Earth's terrestrial hotspots basically have loss of biodiversity from North America up to South America and even the continental Africa, which will be our focus for today. So we do know that Africa plays a significant role in global biodiversity and is also an essential component for the biodiversity hotspots. For instance, we know that the continent of Africa has several biodiversity forms, from intrazonal to open woodlands to semi-desert, and just about any form of biodiversity. And interestingly, in Madagascar, more than 90% of species found in Madagascar are not found anywhere else in the world, but the genomics of the species there are still yet unknown. So we also do know that their human activities poses significant threats to biodiversity, mainly activities such as land and sea use change, pollution, and also overexploitation, climate change, and invasive species and disease, especially as we have seen with epidemics and pandemics recently. So generally, globally, we also do know that the biodiversity loss is happening at an unprecedented scale. For instance, this diagram shows biodiversity loss in percentage compared to an intact ecosystem. And of course, Africa is not left behind here, as we begin to see that the areas in red are areas that used to be that used to have rich biodiversity, but now when compared to the loss there, it's actually changing, it's actually changing from blue to red. And we know what this means, over the next several decades. So there are two revolutionary projects that have and will advance our understanding of human health and biosphere. For instance, over the past 20 years, the Human Genome Project, which was completed the first draft genome, which was completed in 2003, basically helped us to advance and revolutionize our understanding of human health. And the biogenomes, mainly biodiversity genomics, in the next 20 years, will also help us to improve our understanding of the global biosphere. And for the Human Genome Project, we have the HGP, and then for the Earth Biogenome, for the biogenomes, basically we have the EBP, which is driving this agenda globally. So in 2018, scientists basically launched a 10-year program to sequence all 1.5 million known genomes. Actually, there are 1.8 million known genomes now because new species begin to be discovered, basically. So one of the things basically that I started to do a few years ago was when I left my job in Port Harcourt in Shell, I moved to Cambridge. I found out that there is quite a huge difference between the capacity in Africa, mainly in Nigeria, and also elsewhere. So I began to think about a platform that would basically allow us to start advancing knowledge, And then it was in 2015, I got to know about HBioNet, focusing on human health and also doing some non-human species. And then in 2019, I also got to know about Acacia, focusing on plant and also some livestock. But there is something we needed to do, a large-scale genome effort, especially focusing on non-human space, looking at biodiversity. So in 2019, at the Elam Institute, I approached Dr. Rob Davey, basically for us to set up a platform, that would begin to look at this at a continental level. And at that time, we called it DAISY. So in 2020, I basically approached Adesanga, Mark Blasser, and also Harris Lewin in California, mainly for us to connect with the Earth Biogenome Project to set up a continental-wide program. Looking at Africa is very rich in biodiversity, in agriculture, and many other non-human systems, but many of these are not explored yet, and many of these are not being sustainably utilized. So in 2021, we had a first kickoff meeting, basically. So the project is now being chaired by Professor Anne Mugai, because one key thing here with the African Biogenome Project is that it must be driven by the people, by the African scientists, in order to be beneficial to the people. And I think from Shego's presentation previously, we talked about it. He talked about capacity building. I think this is also one key thing we're doing basically within this project. It has to be, the sequencing has to be done on ground in order for the people to benefit from it, and in order to be able to contribute heavily to the people. So the overarching aim here in this project mainly is that we aim to sequence genomes of indigenous and endemic biological species, from bees, just about any species you want. And then at the moment, we're focusing on DAISY, which is a community network, in conservation and agriculture, which I'm going to talk about later. So the idea is that this will be one component of the African Biogenome Project, because one key thing is that it is important that all of this project, come together and get unified, because the knowledge generated in one activity being done by one particular group could benefit other groups. So it is important that, in the non-human space, we kind of pool resources together and bring everyone under one umbrella. And of course, that is what the African Biogenome Project aims to aims to do. Looking at plants, looking at livestock, looking at eukaryotic microbes particularly. So for DAISY, the focus is on food systems and conservation. How do we ensure sustainable food systems across the continent of Africa? And the DAISY network is a community network within Africa BP to support genome-guided sustainable agricultural practices. And the idea is that we're moving from sequence all the way to applications and also looking at resilient food systems and well-being. And I would also mention that there are also other communities in Africa, for instance, the African Farm Crop Consortium, which is basically looking at about 100 farm crops. But one of the key things we're doing, and also we're also working with scientists within the AOCC, is to advance into areas that are not yet explored and that would otherwise not be explored by one single group. So one thing basically with the Africa BB project is that we aim to focus on so many species. We're looking at vertebrate species from mammals all the way to amphibians. And we're also looking at arthropod and platyhelminths. We're also looking at plants, algae, protozoa, and fungi. So what are the challenges? The key challenge mainly is to address the goals of the UN Sustainable Development Goals 02, Zero Hunger, and most importantly, ensure improved biological resources across the continent of Africa. And this basically connects to the African Union Agenda 2063, basically. So one quick question is that, is it one of the quick questions that I've actually received over the past several months, is that, is it possible to sequence large numbers of eukaryotic species? So there are communities around the world who are working on this, and many of these communities are either national country-based, or they are regional-based, or they are taxonomic-based. For instance, the Vertebrate Genome Project is taxonomy-based for global, so they are focusing on all vertebrates. The European Reference Genome Atlas is region-based, is focusing on species across the European continent. The Darwin Tree of Life is country-based, focused on species found in the United Kingdom. Now, of course, we have communities such as the 1000 Genome Project in Chile. So there are 5 grand challenges that we strongly believe that will help to deliver value to the African people. One is genomics and bioinformatics technologies for the agri-environment. How do we bring these systems closer to the people? The second is crops and livestock improvement and health. How will this help to solve agriculture and food security challenges? Then the third is genomics for endangered and endemic species. As I mentioned previously, the continent of Africa is very rich in biodiversity. Then the fourth is actually socioeconomics, ethics, and policy. And of course, the fifth is technology development, knowledge exchange, and industry. And all of this revolves around capacity building and value proposition. So in order to test this process, we started to do the pilot project. So, and we believe that the pilot project will reveal strengths and weaknesses. And the idea is to sequence 2,500 species across the continent of Africa in 3 years. And in order to do this, we are working with the Vertebrate Genome Project to sequence at the initial instance 18 species, which at the moment would basically go down to around 6 or 10 species, basically because other species have been found elsewhere. So we've managed to sequence about 2 genomes in South Africa. So basically, so in the next few weeks, we'll be able to communicate the status of the genome, because we need to go through so many processes, like, to ensure compliance and other aspects of the project. So we are also working with the 10,000 plant genome project to sequence in the initial instance 100 plant genomes across the continent of Africa. So we've populated the list, and we have the list now ready. So the idea is to carry out an evaluation on the list, mainly focusing on species that have very low genome sizes in order to test this pipeline on the continent in Africa. So there are so many committees. So in the past few months, before March, I think we're just about 7 or 10 people working on this project for nearly 18 months. Then by May, June this year, so we're able to put together the pilot committee members, and now there are over 55 scientists who are actively working on this project across the continent and also outside the continent. So there are several subcommittees mainly. We have the sample collection subcommittee, annotation, IT, ethics, and communication and public affairs. So I'm just going to talk briefly just about one subcommittee, which is the sample collection, because one of the things which we've known basically, so this is also this subcommittee is headed by Verena Ras, who is working with Professor Nicola Moda in South Africa. So one of the key things basically in this subcommittee is that the sample collection is the biggest bottleneck. How do we get the samples, and also getting permits for the samples and so many other things. So, but this committee have been able to draw some documentations on how we can navigate some of these things, because you need to go into remote areas to get the samples and preserve them in a way that is suitable for sequencing. Then, over the past few months, we've been able to set up the project. Mainly, as I mentioned previously, is now being moved to Kenya because it has to be led from within the continent. We've been able to set up a website. We've been able to carry out three public engagements. So we have the biodiversity workshop in 2020. We also had it in 2021. In January, there is an Africa BP workshop that will take place in San Diego, California, and we are planning the first annual African Congress on Biodiversity Genomics now to bring together policymakers and continental bodies to this. So we've also secured partnership, with about 17 institutions who have signed the Africa BP MOU, about 3 industry corporations, and also 5 associate projects. And of course, we are working with the VGP and 10KP in the initial instance to sequence some of the species that they are interested in. So again, this is the slide for the 2 meetings. So I mentioned previously that one biggest challenge is sample collection. And of course, within that is also the issue of Nagoya, compliance, monitoring, evaluation. How do we comply with some of those things? So in order to help with this process, the first thing we did basically was to, this month, is to start having monthly meetings with the national focal points on Nagoya and access benefit sharing, to ensure that the communities basically are able to get connected to this project. So, and additionally, we've also had that the foundation of genomics, again, has been built by so many communities, for instance, History Africa, IITA, Acacia, and also the African Orphan Corps. So this is the organogram for the project. So we have the steering committee, which I'm its co-chair, and also we have the advisory board, the partners and collaborators. And we also have a node-based structure. We have the regional nodes and also the national nodes as well. And these are members of the steering committee, basically, who are basically mainly based, 90% are based in Africa. Then we also have members of the advisory board, consists of Professor Nicola Moda, Rob Davey, and also Simplice Nwala at the African Union. So we also do have a website, so please feel free to go check out the website, the africanbiogenome.org, and just to have more information about the project. And you can also check us out on Twitter, LinkedIn, and also other platforms as well. So you can as well send us an email, and there are over 450 email subscribers to this project. So you can as well follow us here as well. With this, I would like to thank all the members of the Africa BP community, mainly the Africa BP committees. So there are over 55 members, as I mentioned previously, who have been helping with this initiative, and also the institutions, the partners within Africa BP. So with this, I would like to thank you all for listening, and I am happy to answer any question you may have.
Awesome presentation. Thank you so much, Ms. Dr. Thank God. So much. Okay, we will entertain questions. I already have a couple of questions down, noted. Do we have any question on the chat? Let me just start. Okay, I see the first one from Tony Ademola. Thank you for the presentation. Just out of curiosity, the DC project that you mentioned, the DC, the IASEA project, do you have a plan to handle product delivery that ensures some level of knowledge exchange and transfer to individuals like the farmers that are involved? How is this translated to local farmers using this group? As just one of the examples.
Yeah, so in one of the slides, I did mention that we have 5 grand challenges. One of those grand challenges is knowledge exchange and industry. So one of the key components of Africa BP is how do we bring these things closer to the people? How do we get it, direct to the people? And in order to help address some of these challenges, within the knowledge exchange and industry, we are proposing for the formation of the Africa BP Open Institute. So that would basically house some of this knowledge exchange and capacity building in order to be able to transmit this knowledge to the people, to the stakeholders, the farmers, the policy makers, and the people who are on the ground, basically. So that is one key area that we are basically taking into account in order to deliver on this.
Okay, thank you. Just a follow-up on that. So as the Ministry of Agriculture involved, have you had an engagement with them in terms of this knowledge transfer? Because they should have an existing structure for that. Yeah,
so we haven't, the project we just had a first kickoff meeting this June. So we have several, for instance, the Ethiopian Biotechnology Institute, they are now part of this project. So we have several government platforms actually coming together. But we understand that many platforms or government agencies is not going to happen at the same time. So it's going to happen, people will build up momentum at different rates. So that is one of the things we expect. But yes, these are things we all welcome, and we have people contributing to it.
Okay. Okay, so there's another question here. Someone is looking at your projects assist in the development of drugs from the African biomes. Can it drive the development from there?
Yeah, so we believe that the knowledge generated from this project will be applicable across board, in drug discovery, in just about anything, because imagine sequencing species, plant-based drug platforms, and knowing what are there, because more than 70% of species in Africa, especially plants, are not yet characterized, let alone sequenced. So we believe that this will be very beneficial when we begin to know, the characterization, the sequencing, and have the full genome. That will help us be able to design some plant-based drug system, and also in animals as well, and eukaryotic microbes.
Yeah, I agree with you. So I'm looking forward to the time when we'll be able to get the knowledge of how the synthetic process of phytochemical synthesis in plants works, and that's I think we're on the line. Okay, any other question? I'm trying to look at the chat. So the organizers, please take note of the questions of people who want to contact the facilitators. So please, the organizers will attend to that, please. Okay, so my question is, in terms of the sequencing of some of the samples, how did you get the funding and the collaboration sorted out to get some of the things?
So one of the things that we do is that we have about 3 possible funding streams. The first funding stream is the supplementary funding. So these are funding coming from partners who have funding already to do the sequencing. So if they have funding to do the sequencing and of this species, and these species are found in Africa, so we work with them to sequence the species, basically, and those are the funding within the pilot project. So the second stream of funding, so of course, with that funding, it resides with the partners. It does not reside with Africa BP. So with the second stream of funding, basically, is that we are also aiming to do similar things with partners in Africa, not just partners outside the continent. So the first and second stream are kind of have some balance there. Then the main funding, which I think is what you're asking about, is the core funding. So we are planning to onboard this onto the African Union, and also we are also planning to apply for a grant. So which we'll be having the meeting next week Tuesday, to understand if this is feasible, first of all, if this is something that will go through such grant, basically, and then that would also help. So we believe that will build part of the core funding. And in the future, due to how ambitious the project is, we know that this project is not a project that will be delivered based on grant application only. So we say philanthropy and things like that, because we are talking about a huge project, in order to deliver on this. So all of those things will come together, actually. Yeah.
So I'm just thinking off the top of my head. So we have this company selling these sequencing machines and the rest of them. Have you been able to look at engaging with them and using their system to run some of your sample as a way to validate their own equipment? And just
yes. So we have Pacific Biosciences in California, and they have an office in South Africa. We have BioNano in the US, and they have an office in South Africa. We also have one other system. So one of the things we've also found is that most sequencing platforms are happy to do this, in a joint effort to deliver on the capacity. And we've seen that, for instance, in the first 2 species that we sequenced, where we also have the sequencing companies coming to actually support the process. While we know that would help to advance it, it's okay when we do it for a few species, but we're not sure how long that will last and how long they can support that. They can, of course, knock off a few percentages here and there, for a couple of species, but we're talking about 100,000 species over a 10-year period. So again, but it's actually good in the first stage to see that they are coming on board, and it's actually good to see that this is being supported from that angle. But for sustainability rather, it is a project that we want to anchor onto the Africa Union or the Africa Academy of Sciences, because it has to be driven from that platform for it to benefit the people in the long run.
Thank you so much. So a couple of more questions on the chat. So somebody wants to know, do you have plans to implement this in humans? I guess the person is looking at the microbiomes in humans and see that kind of interaction.
Yeah, so I think the project is likely to generate so many questions and so many knowledge. And one of those is, for instance, how does that feed back into the human population? How does that feed back into the human system? So one of the things I do know is that proper biodiversity conservation has a huge role to play in epidemics and pandemic preparedness and prevention. Because if we conserve the species very well, if we don't interact with the species, if we know the genetic makeup and the traits, then by doing that, we can prevent epidemics, including epidemics like COVID-19 or just about any other pandemics, basically. So we don't have plans to actively do that within the project, but what we have plans to do is to actively generate the data that will inform those decisions and those contributions, because we are already seeing some other health system. For instance, we're working with H3 Africa. They are the H3 Bionet rather. They are actually a partner in this project. So we generate such data, and then scientists within the health system can then take the data to make any informed decision, basically, that I wish to make. But definitely, the data, there will be generation of data to help in that process.
Okay. So just 2 questions before you go. Thank you for your time. So when somebody wants to know how you this wonderful research, thank God for all the people on the platform, they understand what genome means. How do you communicate this to the public? Do we have a platform? And I think this is one thing that also goes to the organizers. Do we have a platform for that? That's the first one. And the second one, another person wants to know, particularly, let me just look for that. In terms of your data collection, he assumed that you're involving Nigerians as part of it. But in terms of the data you're using, do you see some of them being developed within Africa?
Yeah, so sorry, I think you are breaking off the last question.
The last one. Yeah. The last one was talking about data collection, and we want to know if some of the equipment you're using, do you see them being developed within Africa?
Yeah, so one of the challenges, in terms of doing this sort of work in Africa and by Africans, is that the technology does not reside with us. Right. So you always have to bring it, So you can count maybe 1 or 2 places that actually develop this technology, and that could be in South Africa, and that could be also reagent manufacturer, maybe perhaps not so much the actual core development side of things. So in the future, we hope that this will motivate that to happen. We hope that this will drive this, and which is why, because one of the things we think is that once these things begin to be sequenced by the scientists in Africa, then that would provide the stimulation to actually start thinking about, developing it. Let me give you an example. It's in order to sequence in Africa, it costs much more than sequencing in Europe and the US. It's very clear. The machine has to be shipped to Africa. You have to do the custom clearance, the shipment cost. The reagent also has to be shipped. So all of those costs build up, into what you actually pay in sequencing in Africa. But then you might think that actually, if we manufacture these things in Africa, then we knock off the sequencing, the shipment cost, we knock off the custom clearance, we knock off all of those things, bringing down the cost. So we believe that one of the things we want to do is to create that stimulation so that scientists will start thinking and helping to advance this. And we believe that by doing this, sequencing in Africa, that would also help with that in the long run. And of course, it's embedded within the 5th Grand Challenge, technology development, knowledge exchange, and industry.
Thank you so much for your time.
Welcome.
We appreciate your time. And we hope that more grace and success in your ventures.
Thank you for inviting me.
Stay in touch. All right. Okay. Is there a coffee break, organizers? There's no coffee break.
No, we just end by 1.
Oh, okay. All right. And some people might need coffee. You can get coffee in the middle of the presentations if you want to. All right. So we're going to move on to the next presentation. Presenter will be speaking to us on natural product anti-aging biomarker discovery using RNA-seq, Dr. Alafi Tayo Akinola Adekoya. Okay, and then just introduce you to him. I hope I have it. So.
Dr. Zubike, could you make Akinola co-host?
Yeah. Dr. Moses, do I have it?
Yeah.
Okay, because I'm looking for it. Just a minute. I have to navigate between this platform and another platform. All right. Is already on the platform. If you can reshare it, I will appreciate it because I can't seem to find it.
It's already sent in your WhatsApp.
Yeah, already.
Yeah.
Okay, that's what I'm trying to open.
Yeah, it's in your WhatsApp. The very last one. Yeah.
Okay, thank you. Okay, let me just do this. My apologies. Just give me a few minutes. Thank you, I have it now. Thank you so much. Okay, Dr. Akinola had his first degree in biochemistry from Hamadu Bello University, Zaria. And then he proceeded to Kebbi State for his mandatory National Youth Service Corps, where he was adjudged the second best corps member for his batch. That's good. And landed him an automatic job as of his choice in the state. He lectured briefly at the Waziri Umaru Federal Polytechnic, Benin, Kebbi, before proceeding to Malaysia for his master's program at University Putra, Malaysia. Due to his hard work and excellent academic performance, he was granted accelerated transition from master's to PhD in genetic engineering and molecular biology, and he graduated in 2017. His journey into the world of bioinformatics started during his PhD program, where he applied RNA-seq techniques to study the anti-aging effects of curcuma xanthoizer. Apologies. He also worked briefly as a postdoctoral researcher in plant biotechnology laboratory and Faculty of Biotechnology and Biomolecular Sciences in the same university, where he used the same technique to study phytomediation of arsenic in mimosa, which is another plant, before proceeding to Canada. He's currently a postdoc fellow at the discipline of genetics, Faculty of Medicine, Memorial University of Newfoundland. At the Memorial University, his research focused on multi-omics analytics. I like that. With a particular interest in integrating genomics, epigenomics, microbiomics information to decipher pathophysiological mechanism. I want to welcome to this conference or symposium, Dr. Akinola for his presentation. Thank you, sir. You can proceed. Is he Monsieur the host already? Hello? You've been asked to unmute.
Okay, good morning from this side. Can everyone?
We can hear you. I can hear you.
Okay, all right. Thank you for giving me the opportunity. Thank you for the introduction. As you said, my name is Akinola, and I'm currently a postdoctoral fellow at Discipline of Genetics at Memorial University. This morning, I'll be talking on one of my research where I applied bioinformatics techniques, which is RNA sequencing in natural products, especially in anti-aging research, to find out how we can actually apply this to our work. Actually, my background is actually wet lab, and then I'm coming into bioinformatics. And then I think this is a good avenue to share one of the first work where I actually applied bioinformatics, which is RNA sequencing techniques, to my work. And I know this will actually be useful because most of the research papers coming from Nigeria are more or less like natural product, but we actually try to do extract and then we test on rat. But at the end of the day, most of those researches don't go beyond the shore of Nigerians. And why? Maybe it could be as a result of experimental design, or we're not using the right technique. So since the sequencing of the human genome, we've been equipped as a wet lab scientist with a lot of tools, resources from bioinformatics, which we can actually apply that can actually add values to our research in Nigeria. Sorry. So today, by overview of my talk, I'm going to introduce the topic and what I'm going to talk about. Then talk about natural products, which we all know, and then the market overview of aging products, and then what is actually skin aging, and then what is the mechanism of skin aging, and then how do we bring the RNA sequencing to skin aging research, and what about biomarkers, and then some of the materials I use and my methods, and then I'll talk briefly on the results and discussion and then acknowledgement. And this is going to be totally different from from the 2 or 3 speakers, because I'm just mainly focusing on my research. So from introduction, I would know that Nigeria is actually rich in a huge biodiversity, and Nigeria is ranked among the top 50 countries in biodiversity, and about 8,000 plant species are available, and then we have about 338 families and 2,215 genera. So this tells us Nigeria is actually a wealth of natural products that we can actually tap into. And if you actually harness that, it will actually boost Nigerian economy. It could actually create more jobs and makes our economy to be one of the competitive economies in the world. So now natural products, the world is actually tending towards natural products now. because of the side effect of some of the present in substances we found in the product we found on the shelf in the market. We know most of those care products, some of them have what we call hydroxyl acid, which are actually sunburn and can cause malignant. And we have limonene, which actually can cause carcinogens. And then some of them are actually embedded with paraffins. So all these are some of the disadvantages we have in current anti-aging products in the market. And then now the market, the cosmetic industry or the personal care industry are actually looking towards natural products to see how we can make things safe and how the product can be effective and decrease any side effects. But the issue with Nigerian natural product research, there are no really scientific proof to actually support their finding or to support their claims in what they say. You can say, oh, my compound or my substances can cure diabetes or whatever, but do you have any scientific report or any scientific research to show that this works? So as a result, these are some of the tools we can actually use to apply to our research. Now, the global anti-aging market, let's tell you the economy of the anti-aging market, how huge it is. The global anti-aging market is estimated to worth over almost $200 billion US. That was as of 2019. And then it has an annual growth rate of 7.8% from 2019 to 2019, according to TMR report. And then if you see that the Asia Pacific market is estimated to worth more than 70 billion. So this is a huge money even for a country as you know, on its own. And Nigeria is actually endowed with a lot of product we can actually tap into. It might not necessarily be anti-aging product. It could be anything. It could be medicine. It could be nutraceutical or anything you can talk about. And if you see, according to the trends, the trend is actually increasing geometrically. And if you see Africa and Middle East, we in the cosmetic global industry, we are still less behind. We are far behind because about 4% of our revenue is being generated. So we still have a long way to go in our natural product research. And now to come down to the African markets, with African beauty and personal care, what estimated amount of 6.93 billion in 2021, and currently between 8% to 10% each as increment from previous years, and it was estimated to reach about 10 billion in 2017. That was there. Then the Nigerian beauty and personal care market, that's the country we are in Nigeria, is currently worth an estimate of $3 billion according to Euromonitor International. And this is even more than a state revenue or a state budget. So this is a huge amount that if we can actually go into. So now the race is between how in the cosmetic industry, how to turn back the biological clock, which it seems sometimes it seems impossible. But sometimes when you look at science not is impossible in science as well. Now, mostly what we do is see how we can delay the processes, even though if we cannot reverse it totally, then we can actually slow the process as man's quest for immortality is increasing every day. Nobody wants to die. Everybody wants to be young forever. So, because the cosmetic industry, so their desire to slow or to desire is to either to slow down or prevent the aging process. And this is a very arduous task for them to accomplish. Now, I will take you back to what is skin aging. Skin aging is actually an inevitable process which actually occurs in all human skin. Either you like it or not, you must age. And then it's one of the main features is characterized by loss of skin structural integrity. And then it's characterized by the loss of cellular matrix, which is ESM degradation. And there are two types, which is intrinsic and extrinsic. The intrinsic one are the normal one that actually occur slowly and gradually, which is a chronological aging. And the extrinsic one is the one that is actually called the premature aging, which is as a result of external factors, which could be as a result of excessive exposure to UV radiation, either from the sun or other chemicals, like when you smoke, chemical exposure, and which all this process actually led to generation of reactive oxygen species, which is a main culprit in aging process. So now, let me just take us to the mechanism of skin aging. Usually, if you are hit by the sun, what happens is that the sun contains what we call UVA and UVB, which are rays coming directly from the sun, and this will eat us on our skin. Once it eats us on the skin, it generates ROS. And once ROS is generated, then this initiate the growth factor receptor, and which are the two growth factor receptors, cytokine receptor and the growth factor receptors, and then leading to activation of a pathway called the MPAK pathway and GNAK pathways. Then activated protein are being activated. So there are two parts to the activation protein, which is the C-force part and the C-jun part. So the C-jun part, which is the one that actually responsible for production or transmission of a MMP, which is a metalloproteinase 1 transmission factor, is being switched on. So what happens is that these metalloproteinases will break down collagen, and once collagen is broken or degraded, the wrinkles actually comes. That is one of the pathways. Then the other pathway is inhibition of the C-force part, which actually leads to down regulation or the lack of production of TGF-beta, and that leads to procollagen not being produced, and that also leads to skin aging. And then another part is what we have for the elastase, collagenase, and those are enzymes that actually break down elastin, collagenase, which give skin their structural integrity, like collagen, elastin, this elasticity, and the hyaluronidase, which actually hydrate the skin. So also, as ROS actually binds to the cytokine receptors in the GNK, and it's release expression of these particular enzymes, and they produce and they break down skin structural integrity. So it goes on and on that way. Then even our mitochondria in our body system is actually, they generate ROS as well, because as we breathe, as they produce energy, they actually release ROS, and that ROS they produce. This is one of the major causes of increased aging. That is what we call the chronological aging. That is not actually, we can control. And that also leads to formation of lipid peroxide, DNA damage, and also skin aging comes in. So now, let's go to RNA sequencing. So RNA sequencing is actually a next generation sequencing technique. And what it does is that it reviews the presence and quantify all the RNA present in the biological system at a particular point in time. Now, it captures the total RNA in the cellular content, either mRNA, ribosomal RNA, or transfer RNA. And then these techniques actually could be applied in areas for like identification of new disease biology, biomarker profiling for clinical indication, which is one of the areas I'm going to talk about, genetic diagnosis, and also it tells us the molecular mechanism of potential therapeutic target of natural compounds. That is, we can actually establish a mechanism of how a particular product, a particular drugs actually works via RNA techniques, using RNA techniques, and then we can identify genes related to a particular treatment at a point in time. So the advantages go on and on, the application goes on and on and on. So this is just a few one for this purpose of this presentation I'm just talking about. Then what are biomarkers? Biomarkers are actually, Okay, biomarkers are actually, they are substances that can actually use to measure a normal biological process, either when, for example, if you have like a treatment and you want to see the efficacy of a particular extract or a particular compound or drugs, you can use biomarker to actually monitor that time, the time of exposure or an intervention. And then they also ranges from biomolecules such as, it could be carbohydrate, it could be lipid, it could be gene, it could be DNA, platelet enzymes, or whatever. It could also be metabolites. And then sometimes they help in identification of a particular disease, which is, I've talked about that earlier. And then biomarker, one of the characteristics that they can be objectively measured in a system. So they can tell us, they can be used for personalized medicine. They can tell us, okay, by the time you start seeing this biomarker, it's a sign that maybe you're having this disease or you're having this problem, or in the next future, you may be having this, so you need to be careful. So they could be genetic. So in my project or in my research, what I did in my PhD research, what I did was I actually work with some plants, and this plant is called Zingiberacea, and they are common all over in the tropics. This plant is known as ginger family, and they are used as spices in our food. They are used as condiments, flavoring, or sometimes they use them as a traditional medicine. So I work on the 10 of this plant. So what I did was that I first screened them. Okay, sorry. Secondly, I use human fibroblast cell, which is developed or gotten from human, a 34 years old Caucasian, and it was gotten from the abdominal. And why the choice of fibroblasts for these studies was because fibroblasts are actually present in the dermis of the skin, and they produce collagen, which actually give the skin the strength and the resilience. And then they're actually made up of 3 amino acids, which is glycine, hydroxyproline, and then any other amino acid to make collagen. So type 1 collagen is monitored by the procollagen formed in the dermal fibroblast. So the method I use is that, first of all, I extracted this plant using different solvents, which is what we do at home there in Nigeria, how we do our extract. Then we're using about 4 or 5 grams of that, and then reflux. Then after that, I assay for flavonoid. I assay using antioxidant capacity for each of them. Then I also assay for FAME, which is fatty acid, using HPLC and GCFID. Then I went ahead to do the self-proliferation, in which I grow the skin cell in the control environment, which is the protocol, according to the protocol. And after that, I got the cells. Then lysis, I lyse the cells, and then after lysis, then I did the counting, then see those cells to grow them before my treatment. And after that, after I've treated the cell, what I actually did was I stimulate UV. I expose the cell to UV radiation. Then after exposing the cell to UV radiation to stimulate the stress and aging process. So I now treated with my extract to actually see if my extract could actually have any effect on reversing or having effect, any positive effect in controlling the aging process in the skin. So what I did was, after growing, I chelated the cell, then I sorted the RNA, then prepared the library. And then the library have about 5 steps of library preparation for your RNA-seq. Then I sent it to sequencing using Illumina, and then I did bioinformatics analysis using searching for this, answering these questions, and I validated my result with using the qPCR. So in the bioinformatics part, what I did was that I looked for gene ontology, which tells me what's going on in the biological process, cellular process, and molecular function. And then I looked at the PEG pathway to see which pathways are involved when a skin cell is exposed to UV, and also when you treat it with my sample, what are the effects? So what are the pathways that are activated? And then I looked at differential gene expression to see genes which responds to the treatment after exposure to UV. So down to my result, first of all, what I did was here, I actually tried to get the minimum, the best concentration which will be suitable for my cell. So I actually optimized ranging from 0.9 to 2.5. I can see that at 1.56 microgram. We have the best one. The retinoic acid was used as a positive control, because it's known as a very potent antioxidant, which can actually reverse the effect of aging. We can actually mop up ROS from the system. But I actually came down to 2 out of the 10 because there are some previous preliminary tests I've done from all those plants. What I did was I had to screen them for flavonoid, antioxidant, DPPH, and FRAP. And then I also checked their biochemical component. I also checked their toxicity. I also looked at their toxicity using zebrafish, and then to see how safe they are, even to check the embryotoxic effect. So I came up with these two, which is Curcuma xanthorrhiza and the Curcuma lunga. So I went ahead to try these two to treat them, with my cell. After exposing the cell to UV at 20 millijoule per centimeter square, what I did was that I saw that Curcuma lunga, they are both potent, right? But they can actually reverse the effect of the UV. But Curcuma xanthorrhiza actually gave us the burst. So I went ahead to use Curcuma xanthorrhiza for my RNA sequencing objective. So these are actually the pictures of the cells. Here, this is a morphology showing us that the ATTC cell line of the human cell line treated with the retinoic acid after 24 hours of culturing. And then here we have the cell that has been treated with what we call it 20 millijoules and then treated with Curcuma lunga. And then we have here the cell that have been treated with Curcuma xanthorrhiza after being exposed to UV. And then here is a cell that actually was not actually treated. And we can see that we can all see that the cell's morphology is not as good as those previous ones. Like I said, the cells are already dying. And this is just the control without any treatment, without anything. That is without retinoic acid, without any of the extract, without being exposed to UV. So in my result, I also have, so after doing the RNA sequencing and then checking the differential express gene, I was able to find that in the UI means UV irradiated cells without being treated, that we have about 2,000 genes that are actually upregulated and about 3,000 cells that are downregulated. And then for the one that are actually after exposing to UV and I treated it, then we have about 2,284 cell gene upregulated and then 2,009 downregulated. But my interest lies in here, in which the uniquely differential express gene in such a way that after being exposed, they are upregulated. But when we treated it, they are downregulated with the sample. So that's give me like an overview, okay, my extra which is treated, those are the genes that are actually responding to the treatment in the system. And then I have here what we call the differential experiment gene for Kano plot. So the reds one are the one we're actually interested in. So which are mostly the one uniting this on the highly expressed genes. So these ones are the ones that actually they could be potential. They could actually be potentially serve as a biomarker or whatever we want to use them for, or as a drug target. So we have the list of the genes here. So these are the lists in this panel we have here, we have the heat map representation showing the differential express gene for each of the cell of the treatment. The one we actually, the UV radiated one and the UV treated radiated one. So we have a couple of genes that actually are candidate genes, which are our genes of interest, which we're going to take further to study. And these genes could actually be like a genetic marker to tell us or to show us that there's a premature aging going on, or when the target for the treatment of skin aging. So the pathways that are actually, after that, we went to study the pathway analysis. In the pathway analysis, I think there are two panels, sorry. So the pathway analysis, these pathways we call them cytokine receptor interaction as a result of UV. Here, this pathway that is one of the pathways that actually was upregulated. And in such a way, after we treated the cell with UV. So, and then we can see that this is basal carcinoma pathways, which shows us that the knockout of some genes, like the P53 gene, which is oncogene, which is gene, what they call it, tumor suppressor genes, which has been knocked out. And that can give rise to skin inflammation, skin cells, skin cancer. This pathway was actually the one that was actually upregulated, or one of the most upregulated pathway. And then after treatment, this is T2, we have the MPK pathway, secondary pathway, showing us the reversal of the pathway. Now, these pathways are actually, we treated the sample with, we exposed the sample to UV, then we treated with the plant, with the extract, and then, so from this one and this, we can see that this is actually a pathway that leads to cancer, and this is actually a pathway that actually leads to reversing of the previous one. And we can see that in the pathway we have P53 pathway being activated in this way, which is what they call it, tumor suppressor gene, to counter the effect of this. So this tells us that, okay, the extract is actually having a positive impact or effect on the UV process. So the findings. Sorry, I can't see this. Okay. So now to summarize the findings, what we did was that we reviewed, the RNA-seq reviewed several differential genes that is being expressed in both UV radiated cells and treated with extract of our saccharizer. And then we went ahead. The key another findings is that the KEGG analysis pathway shows us the induction of some UV-induced pathogenesis pathways, such as cancer pathway. And then we also find that MPK signaling pathway was induced in the saccharizer extract treated with UV in human dermatofibroblastic cells, showing the mitigating effect of this first pathway. And these findings has implicated UV as an agent of premature skin aging and provide clue for management and also treatment. So after I got my gene of interest, so because the gene were actually identified using bioinformatics tools, so I need to verify, I need to validate this gene to see if they're actually true, or if they are not, or is it that, are they fluke or are they there? So I went ahead to do what we call quantitative PCR analysis of the gene. But I didn't do all the genes, I just picked 10 genes, 5 from the upregulated one and 5 from the downregulated one, to actually see the effect in real life. So so these are the list of the genes that are actually highly regulated. And then these are the ones that are highly down-regulated. So I designed a primer, and then these are the NCPI sequence ID. So what I did, I grow the cell that was treated, extracted RNA from these cells. So and then this is actually the the gel showing that the RNA was actually extracted. And I converted that to cDNA. After converting to cDNA, and then run the gel to see that actually they're actually present, and that these things are, the cells are present. And after that, I ran, I will call it quantitative PCR to check the expression, to compare my qPCR expression with the expression profile I got from Bioinformatics. This is just to validate. So I noticed that the result shows that the RT-PCR gene expression analysis, so that these genes are significantly upregulated in the UV predated human dermal cells, but they are downregulated after treated with xanthorizer. So which means when we expose them to UV, they are upregulated, but after treating them with the extract, they are downregulated. So which means they are being controlled by the extract. And also, these are another set of genes which basically they are downregulated in the UV, which could be as a result of the UV. And also after being treating them with the extract, they are upregulated. So these are the likely candidates genetic biomarker that we could use as to monitor the treatment, and also it could also be like a sign for personalized treatment or detection, or sorry, to detect if you're actually undergoing premature aging or not. But this is not really a problem in the Black continent, most like that, but we still have it there. But it's more prone in the white countries in where they don't really have melanin. And because of that, when they're exposed to too much sun, they age very fast. So in conclusion, the following set of genes, that is this set of genes were found to be significantly upregulated in the UV human dermal fibrous cells, and they are downregulated after extra treatment. Why these other ones were actually the opposite side, opposite one, they are downregulated and then upregulated after xanthorizer treatment. Therefore, we now concluded that this set of genes there will be a good potential genetic marker or biomarker for early detection of UV-induced what we call premature aging. They could also serve as a marker to show the exposure rate of UV. If you've been exposed to too much UV, if those genes are highly regulated, you can measure them at a different point in time. Then you know, okay, I'm getting too much exposed to UV. I think this is common in a country like maybe like Australia and some other countries that actually they have white, but they have each sun rays over there. And then, so one of the products that actually came out from this was this proposal. Before I left Malaysia, they were trying to, in the lab where I was, they were trying to develop a anti-aging diagnostic kit over there. So using those set of genes. So, but I think someone else is kind of taking over the research because it's need to continue, because they need to fractionate those extract and see which compound is the bioactive component that is responsible for this effect. And also they need to actually test this to look at those genes on the clinical level, on the real human being. Even though we are using cell line, this needs to also to translate into, for us to translate into a real damaging kit, we still need to do further research. And then another one also that also came up is this Xanthodermal anti-aging body cream. This is so these are ways in which we can commercialize product, because Nigeria is actually rich in abundance of natural resources, but how do we make good value from these things? How do we make money from it? How do we create wealth for the nation? So moving forward is that we have to look for ways to make sure we standardize our research to see how our research could meet global standard in such a way that when we have an experiment or we design anything, we should be able to back it up with real scientific findings. By so doing, that will give us the opportunity to hit or the global market, the huge global market in drug discovery, in nutraceutics, in anti-aging, in whatever areas of our research. So I would like to acknowledge University of Putra, Malaysia, where this research was conducted in the Plant Biotech Lab. And then API, where we used some of their facilities, and then Syngenics, where the sequencing was done. And then Aragene, where we did some analysis, and also my current place, which is Nigerian University of Newfoundland. All right, thank you. So I'm open for questions with regards to the research.
Thank you so much. And I mean, I can already see on the chat the appreciation of the in-depth work that was done, well-designed work that you did. Really, we can have we only have room for one question, and I already have some question on the chat. So I'm going to combine two questions to you. So somebody want to know, do you have plants already that have been tested like this and that have this kind of data that you generated for this plant? And another question just to add to that was the issue around telomeres, this repeated unit at the end of chromosomes. And we know that they have been linked to aging over the years. Was there any way to monitor those sequences in your research? Or can only take those two things? Okay,
response to the last one I was talking about telomere shortening. Okay, so telomere shortening is as a result of division of cells. As the cells divide, then they keep on losing a portion of the telomere, which is actually attributed to aging. Now that is what we call chronological aging. So that is like inevitable, but we're looking for a way to slow. Our focus here is premature aging. Aging that's as a result of external factors that are a result of maybe exposure to chemicals, that are maybe exposure to sun rays, okay, which contains UVA, UVB. So these are the effect of what we're actually looking at, not really much on chronological aging. If you in my slide, I said that it's irreversible. With the scientific community just trying to reverse aging through the process of what we call the second man quest for immortality. So yeah, I haven't been answered yet. So that is that I not. And then the second question is those plants, right? Can you read back? Like,
so if you have looked at any other plants or there are data for any other plants?
No. Okay. What actually formulates those particular plants? Those plants are used in folkloric medicine. Before now, they are used traditionally, medically for anti-aging process, but they use them locally. They will just crush them and rub them on the skin. And they said that, they actually have effect in renewing the skin or reversing aging. So those 10 families or Zingiberacea are the plants they use in Malaysia for anti-aging process. So that was what gave us that idea, that look to move towards those plants. We have not actually looked at any other plant, because looking at plant generally without direction may be like waste of money, waste of resources. So we work based on previous folkloric medicine, natural products, so for us to go towards those plants.
Okay, thank you so much. So just do us a favor. If you have a publication on this, if you can put it on the link on the chat, it would be nice so that people can get.
Yeah, I have about 2 or 3 publications on the where we actually one is okay. I will. Do you want me to drop them on the chat?
Yes, please put the link on the chat. I'm going to be quite helpful for so many people.
Okay. All right. Okay. I will do
Thank
that.
you so much.
Thank
you so much.
Yeah.
We appreciate your presentation. Thank you once again, Dr.
Sorry, I didn't open my camera for everyone to see me. I think before I go.
We should see this face. I didn't say.
yeah. I forgot. So can everyone see me now? So the background, I can't see myself anymore. But the background you guys are seeing, that's the city of St. John's. Yeah. So thank you for watching.
Okay, I'm looking at your background.
Yeah, the background is actually the city where I am. That's called the city of St. John's in Newfoundland.
Oh, beautiful.
Yeah.
Beautiful. Nice to see you, sir. I appreciate it. Thank you so much. Okay, we need to move on quickly. Very exciting presentation indeed, I say. Okay, we're moving on to, I believe, another exciting presentation by Dr. Chinwe Ekena. She is an assistant professor in the Department of Computer Science at the University of Albany, State University of New York, and the director of robotics. Oh, there to my heart. Algorithms and Computational Systems Laboratory. Professor Chinwe's research centers on intelligent motion planning applied to robotics and proteins. Wow, she has explored intelligent adaptation of robotic motions planning to improve planning time, topological data analysis methods to capture important features of robotic planning spaces. Our research interests includes machine learning, computational geometry, and computational biology. Professor Chinwe is a recipient of the NSF CRI Award on Topology-Aware Configuration Spaces. And she has gone to publish several works on the BIBM. We'll get to know some of these acronyms, ICRA and IROS on this subject. She's currently an associate editor of the ARAEL, and has served on several program committees for the same ICRA, IROS, and
WA. Host is coming in.
She's a committee member of the IIERAS.
I cannot now. If I should I be worrying you?
Okay, now I can.
Don't even have that option to do that.
Okay, now the organizers are getting into my space. Okay, she's a committee member of the IIERAS committee to explore synergies in automation and robotics. And she comprises top researchers in the field of automation and robotics. I'm particularly interested to look at the protein motions that might be in this particular presentation. So, please, let's welcome. Have we given her the right to share? Do you have the right to share? Okay, she does. Thank you. You're welcome. Please ask her to unmute also, if you can. The right to unmute, yeah.
Give her right to share. She's off again.
Okay, she's off again. Please do that. I can't see her on this list. Right now. Can you hear us? If you can just speak, we can, so that we know you're muted.
Okay. Can you hear me now?
Yeah,
perfect. Yeah, thank you.
Okay. Thank you. So do you have access to my full screen?
No, I have your the other presentation, not the presentation mode. So if you want to put the presentation mode.
Okay. Times when I have...
if you can.
Okay. Just give me a second.
Okay, you're using double screen.
I have too many screens on. So
yeah, I can see the screens.
I have this issue. I need to confirm where I am. So just give me a second.
Okay, that's fine.
Yeah. Okay. So do you see my screen now with?
Yeah. So you just need to play on the full screen, and we will be fine.
So what do you see now?
Okay, so you're still playing with the current slide and the next slide in the picture.
And the next slide.
Oh, so okay, it's fine. You can just put it up without going to full slide, and you can just move on your side, and that is a better view.
Okay.
She seems to have lost coach host. Could you host? Could you share now, Chinwe?
Okay, give me a second.
She lost host again.
She lost the coach.
No, I stopped the share, so just give me a second. I'll go figure it out now. Yeah.
Okay.
So we're good now.
You're good. Okay. So yeah. Oh, beautiful. Cool.
Okay.
Okay.
All right.
Thanks.
Okay. So thank you very much for inviting me for this talk. So I know we looks like we're really out of time. So I'm just going to try and just browse through some certain aspects of the work, but then really delve into some very important aspects I want to give as a takeaway. So my name is Chinwe, and I got my BSc and my MSc from Covenant University in Nigeria, actually the first set. And then I moved on to Texas A&M to get my PhD. So the image I have there is with me and my advisor, with some of my PhD colleagues. So the interesting thing you have there is you have people who went for a conference, and then we have people who have graduated with their students, and then we all met at the conference. And then the lady in the tie and dye is actually my advisor. She came to Nigeria for a conference at Covenant University. I got interested in her research, which led me down, I call it a rabbit hole, because I was working with Professor Debbie, really looking into bioinformatics aspect of my master's project. And then ended up going to Texas A&M University and doing a lot of work in robotics. But my passion has always been the whole computational biology aspect of things. So now I'm at University at Albany. I'm back to doing what I want to do. So I'm slowly getting back into computational biology. But right now I'm still very heavy on the theory and the computer science side of things. So my research interest really right now is like a 50-50 split between robotics and also computational biology. So I'm just going to be talking a little bit about some of the research I've done in computational biology, looking more into structural biology, protein folding, and also give you just a brief about this intersection between what I do in computational biology and also in robotics. So I'll be talking about topological systems. So I brought this image to just give you a glimpse about when I talk about topology systems, we're looking at landscape topology, like the topology of a mountain. This will be what you would expect to see. You'd expect to see ridges, you expect to see valleys. You'd expect to be able to represent that topology in such a way that if someone sees a topology of that landscape, they can give you an idea of what they are seeing. So an urban city topology would be, you expect to see high-rise buildings, you expect to see parks and the rest of them. And then if we're looking at proteins as well, you have your proteins as your 3D tertiary structure. You'd expect to see some surface representation of that particular protein. So I'll be really touching on looking at protein structures, protein folding, and that intersection. So I'm just going to give some very fundamental aspects to protein, which all of you would be aware of. But for me, coming from the computer science side of things, when I need to give talks like this, I always have to give some very fundamental definitions that most of us are already aware of. So if we're looking at proteins, we look at protein molecules as being complex three-dimensional structures that look at the folding of a sequence of these bases. So if we say bases, we're now looking at the primary structure. So we have the sequences of our amino acids, and then each of these bases being represented by some alphabet. And then you can also obtain this primary structure from your experiments. Then the tertiary structure becomes this sequence loops. When it begins to loops on itself and folds into 3 dimensions, then it now begins to talk about the tertiary structure, just as this image we have a tertiary representation of an RE molecule. Then looking at the tertiary structure, some things we consider really now looks into the chemical bonds and also the potential energy. So I'm really talking about the potential energy because that's basically what we would use in our calculations and how we develop our model. So these potential energy calculations are usually based on these numbers and the types of bonds that are basically developed to classify these conformations. And the lowest energy conformation is usually known as native structure, and then conformations with these few bonds or within these high energy areas are referred to as being unfolded. So with that information, you now go into this folding process, which is usually seen as some black box. So I have this unfolded conformation, which is at high energy, represented usually by these sequences. Then using some physical laws, it causes this folding, and then we have a native conformation in a low energy. So that leads us into really now studying the folding landscape. So you're looking at the folding landscape, that would now have us be able to describe this black box of folding that we talked about. So a space in which every point corresponds to a conformation and its associated potential energy is usually defined as what we call a C space. So in robotics, when we talk about a C space is known as a configuration space or a conformation space. So it's that space of every possible conformation that that protein can actually have as it's going from this high energy state to its more native tertiary structure state. So this complete folding landscape contains a point for every possible conformation of a given sequence. So I have that description here in the image, looking at the native state, going through different variations within its landscape based off the potential energy. So that's one way we make that calculation. Get my slide to move. Okay, so oops. Okay, so these conformational changes actually helps us describe a molecule as it changes physically to fold from one conformation to another. So for the protein folding model, it's really continuous. So your bond angles will change with these continuous rotations that are going on. So really what we say is we perturb the bond angles as it changes from one conformation to the other, while it goes down its folding landscape to its native state. So what are these features of the folding topology? So your folding pathway would consist of a set of conformational changes your molecule is likely to fold into when moving from one conformation to another. So we're going from, looking at this image, we're going from N to X to Y, because we're going down the slope to a low energy area. Then we have these energy barriers, which will be areas of the landscape with high energy that separate groups of conformations. So Y is separated from X and N because you have an energy barrier there. Then we have intermediate states, which are now conformations lying on the folding pathway, which represent local minimum of potentials. An example would be Y and X. So what would our folding pathway now look like? The unfolded is going to just be a set of sequences, almost like you have your string. And in between that, we have these different intermediate states, and then it gives us a folded process. So as biochemists, when you're doing your folding, this happens very quick. But then what we start looking at as computational biologists is, can we provide these different intermediate states that would become of importance to biochemists? Because diseases that are known to cause misfold, diseases that, like Alzheimer's, are known to be caused by misfolding. So being able to produce at every time step what these different intermediate states would be was one of the outputs we were really looking at doing. So there are existing techniques for mapping this topology, but then they really look at these existing techniques I'm talking about are computational techniques. So, but they really look at relatively short sequences, just about 200 nucleotides. But then the robotics motion planning technique, which I'll go into just briefly, called the PRM, has successfully been applied to protein folding to produce a graph of the folding process. So that will lead us to, I'm talking robotics, I'm talking protein folding. What exactly is this relationship that we're talking about? So what we did is, if we're talking about motion planning for a robot, the main aim is you're trying to get the robot to move from A to B. We as humans, it's something we don't think about. If we need to get into the next room, we just move into the next room. But then for you to be able to get a robot to actually do that, you need to plan for it. So that's where we call this whole notion of motion planning. There's a whole lot towards motion planning that I'm not going to get into. But the main thing we really look at is we're looking at validity. What a valid place for the robot to actually move into to avoid colliding with obstacles. So it becomes collision free. So that same algorithm was what was now embedded into protein folding. So in fact, during my PhD, one of my main tasks was, I was in a lab where we had a lot of people doing robotics, and then we were about 3 or 4 of us doing protein folding. So our task really was, robotics, they kept throwing out algorithms. We had to convert those algorithms into bio-usable algorithms. So the proof or the theorem is there that these robotics algorithms, there is a parallel way you could actually use this same algorithms for protein folding. But now you need to change what your input is going to look like. You need to change what you say validity looks like. So validity in this case is now going to be low energy compared to validity for a robot is going to be collision free. So low energy because, based on my introduction, we're looking at saying the native state has the lowest stable energy. So validity would be avoiding high energy areas as it goes down the landscape. So that was the idea behind this work. So I already talked about why we need to study it. Misfolding discussed by misfolding proteins, all of that, Alzheimer's, mad cow disease are related to these misfolded proteins. And then computational approaches would help guide or design future experiments, because now one of my products would be a host of intermediate states you could actually use. So I'll be talking about motion planning. So let me just give you a brief about this whole notion of motion planning, because everything else falls within the realm of motions. So given this movable object, the aim is to find a sequence of valid configurations that moves that object from a start to a goal position. So in motion planning for foldable objects, is given this foldable object, find a valid folding sequence that transforms the object from one folded state to another. So that was the parallel representation we had for all the algorithms we started embedding from motion planning in robotics into motion planning for folding. So the PR method, which is the motion planning method for folding, is we generate nodes. When we say we're generating nodes, these nodes are now going to be these conformation states, different possible intermediate states our protein can actually be in. And then we're doing that upon this topology of the space. And then we now connect to form a roadmap. So you connect if there's an energetic feasibility between each node. So our roadmap now ends up being like a net being laid down on the protein's potential landscape, or the topology of the protein. So the potential landscape would be, we're making calculations on the potential energy for every intermediate state that we get, and the potential energy feasibility for every edge that we generate. So once we do that, now we can now extract multiple paths from its unfolded state to its native state. So that's an example of what we do. And then how do we model the protein? We do almost what we call a quasi representation of the protein. So we're really looking at the amino acid being these pairs of 5 side angles. So the 5 side angles are basically what we continuously perturb to give us these different intermediate conformations. And then to analyze, because now we've done this very interesting work, to analyze this folding potential landscape, we'll now start looking at the secondary structure formation order. Secondary structure formation order, we'll now start looking at alpha helix, beta sheets, when do they fold? Because biochemists, we already, okay, you expect the beta sheets to fold before your alpha helix for this particular protein. So that's something to be able to analyze the feasibility of what we're doing. That's what we basically looked at. So the timed contact map, at what time is there a contact between these beta sheets? Some experimental validation. Then we looked at also studying different folding kinematics. But then I'm just going to touch on secondary formation order and also this distribution for different types. Potential energy versus the RMSD for the roadmap nodes. So what we did here was we calculated this RMSD between each contact as against our potential energy. And then based on their research, realized that these images are formed, like first one is if you have just alpha helices in a particular protein, this is the kind of image we would expect to find in this plot. If there's a mix, this is the image we get to find, and then all beta sheets, this is what we also get to find. So this was one very early way we were able to see what kinds of secondary structure we were actually forming. And using this to also validate. Then looking at different, so we have this list of different proteins that we studied, and then looking at the number of residues that they have, and then looking at the structures that they have, and then basically comparing them with what we have in literature to see if it compares with the secondary structure formation order that biochemists have in terms of we also producing it computationally, the folding process for these different proteins. And then for where there is data available, what we had was basically agreed for actually 4 out of 5 of them. So that shows that we're actually somewhat going in the right direction. So I've talked now about the classical representation of going from motion planning in robotics to folding. It's very parallel. That's basically what we do. We generate a graph. Your graph is going to contain your nodes and your edges. But then one thing we know is, I was also talking a lot about the landscape, talking a lot about the topology. But then if you're looking at graph, those of us that are looking into networks, graph networks and the rest, represents only one dimension of the landscape. But then this landscape is actually multidimensional. There are things in the landscape like your local minima, your local maxima. There's a lot going on in the landscape that your graph will not capture. Your graph is just going to capture nodes and edges, connectivity, and that's it. So one extension I say really looking at now is there a way we could actually study the topology of this landscape? So this is very recent work. And I started really testing this on protein ligand interactions. So if at any time organizers I'm going above time, please just let me know. But I think I'm almost done. So we're now really looking at these protein ligand interactions, because this is now me extending towards my comfort zone, which was proteins, robotics, motion planning, extending towards something also very important. So protein ligand interactions, we all know, are crucial to a wide range of biological activities and functions in organisms. So then your protein surfaces actually contain essential biological information, which would help us understand the properties such as the geometrical organization of biomolecules. And then if you understand its properties, it has life-saving biological